Any questions before we get started today? All right, our game plan is pretty simple and straightforward. We have one last lecture to go. A little bit more histology to do. Time for a Q&A review, all leading up to Tuesday's exam. All right.
I have a quick question.
Yes.
Do you want us to call brush borders and enzymes enterocytes or brush border enzymes?
I would be fine with either.
Okay, thank you.
Yeah, either would be an acceptable way of describing that. Again, as we've seen many times in this class, there's more than one appropriate way of identifying things, and you're usually welcome to use any of those. All right. Any other questions? All right. So we left off last time and we were working our way through our absorptive pathways. We had talked about that there are going to be four main pathways you are going to be responsible for. And the first one we looked at, and I'd like to review it again today, is carbohydrates. So again, we drew it out by ourselves or wrote it out by ourselves, but let's put all the pieces together again, because your book does a good job of talking about this as well, for the most part. Carbohydrates need to be broken down into their basic building blocks, which are what again?
Monosaccharides.
Excellent, and down into those monosaccharides. Remind me again where that occurs. Where is it broken down? We need to break things down two ways. So let's break it down chemically first. Chemically, where is it broken down? Mouth, excellent. How?
By salivary amylase.
Excellent. Anywhere else? Is it broken down chemically in the stomach? Pancreas with the small intestinal pancreatic amylase, absolutely. Again, pancreatic juice, I'd like you to be a little bit more specific by identifying the specific enzyme. You're absolutely correct in that the juice contains that enzyme, but pancreatic juice would be an accurate answer for all of them. So we want to be more precise with those specific enzymes. Excellent. And also in the small intestine, don't forget, as we were just talking about, those brush border enzymes as well. Excellent. All right. Chemically, those are the three places where we break it down. What about mechanically? Where are the places we break it down mechanically? Mouth with mastication.
And segmentation in small intestine.
Segmentation in the small intestine and churning in the stomach. Excellent. All right. So we have those. Right, our amylases in the mouth break down into diglycerides and triglycerides. Those brush border enzymes are going to break it down into monosaccharides. Some examples of these, and again, remember, you don't need to know names of individual brush border enzymes, but one that most people are familiar with is lactase. Right, lactase breaks down lactose and things along those lines as well. So maltase and things along those lines are all there in that brush border, breaking it down into those monosaccharides. As we talked about, we want to get these sugars into the body. It is important to get these sugars into the body. So we use both active and passive transporting mechanisms, where we are going to both use co-transporters, using sodium as the driving force to get that glucose in the apical surface, but we'll also use some carriers for facilitated diffusion as well. Once they're inside the cell, we've stopped them from being lost as part of our, feces at the end of the day. So we don't have to worry so much about wasting energy, not that it'd be wasting energy, but using energy to get them out of the basal surface. So we fully use facilitated diffusion to get it out. Whereas we talked about what is it enter into, what is into that interstitial fluid?
Blood capillaries.
Into the blood capillaries, and then we actually were able to trace that pathway all the way back to the heart. Excellent. All right, so there it is entering into our bloodstream. and then we've traveled to pass back. Again, your book's got some nice examples of, again, emphasizing these enzymes, where they're located, how this breakdown is taking place. And so it does a really nice job of describing this process. All right. So again, we went through that one fast because that was review, because we did that at the last one, but I just wanted to remind us of what we did with that. So any questions on that? Because now that we've done that, I would like to do something new. All right. Anyone? We happy with that? Okay, perfect. Excellent. So let's bring up our whiteboard again, and let's move on to our next macromolecule that we want to talk about. And this is going to be our proteins. Now, of course, as we know, proteins are broken down into what?
Amino acids.
Excellent. Excellent. So we want to break those down into amino acids. And we're going to break them down two ways, mechanically. So mechanical breakdown or digestion. Let's go ahead and do it that way. Where are and what are the mechanisms that we use for mechanical digestion of proteins?
The mouth.
Okay, what do we do in the mouth?
We chew.
Excellent. Although we want to use fancy terms. So mastication, right? Where else?
Stomach churning.
Excellent. Any others?
Small intestine segmentation.
Excellent. Perfect. In fact, if you think about it, these three processes, mastication in the mouth, churning in the stomach, segmentation in the small intestine, they're really mechanical mechanisms for breaking down all of our macromolecules. So really, we'll be able to use these three. We pretty much can write them in stone. They're going to be part of the answer for all of them. Now, with carbohydrates, these were the only three ways we mechanically break down our carbohydrates. However, is there an additional way that is special for proteins and proteins only? Well, I'm asking the question, so the obvious answer is, of course, yes. So let's switch it around. What is the other way that we mechanically break down proteins? Bingo. Alexa got it. Absolutely. In this case, it is in the stomach with that hydrochloric acid. And what does that hydrochloric acid do to the proteins? What would the fancy name that we would say of what that hydrochloric acid is doing to it? It denatures it, exactly. Remember, denaturing is changing the shape, and changing that shape is opening it up, making it easier for the enzymes to get in there and get the cuts. But remember, denaturing doesn't break any of the bonds. So it is not a chemical breakdown. It is a mechanical digestion. And again, this one is only for the stomach. Only for the proteins. Perfect. All right, let's talk chemical digestion, locations, mechanisms, and what produces them.
In stomach.
Okay.
Pepsin.
Pepsin, excellent. And what produces pepsin?
It's the active form or pepsinogen.
True, it is the active form of pepsinogen, but which cells actually make it? There you go, Alexa's got it. Chief cells.
Let's see.
So those chief cells make the pepsin. Excellent. Any other chemical digestion?
Gastrin.
Well, remember, gastrin's a hormone. It's not going to help us. It may make more pepsin. It may make more hydrochloric acid, but it doesn't actually physically, chemically break down. It doesn't break the bonds. Okay, so absolutely, we have enzymes in the small intestine that are produced from the pancreas. One example are peptidase. What is the most common peptidase that we talked about that was made in, came from the pancreas? Trypsin, there we go. Chylotrypsin, carboxypeptidase, absolutely. So all of those are example of peptidases. Their job is to break up the small peptides that are in there. And of course, what else in the small intestine is going to help to facilitate the chemical breakdown of our foods? Brush border enzymes, there you go. Excellent. So there you go. There we have our mechanical digestion. We have our chemical digestion. We have now taken our proteins and broken them down into amino acids. And that then brings us, of course, back to our cell. So let's cheat, make a big block-shaped cell like this. Go ahead and put some microvilli on the outer surface so we can tell apical from basal. Let's make this smaller so it fits nicer. Proteases are basically, proteases are enzymes that help to break down proteins. Peptidases help to break the peptide bonds. They're basically two names for the same types of things. All righty. Now, our goal is going to be to get those proteins across the apical border. Because remember, if it stays out here in the lumen, it's going to be lost from us. And as we remember back to 430, how important are amino acids? How important is it to get new amino acids so we can make new proteins? How important is protein synthesis in a cell? Absolutely, super, very, very important. So we want to get as many amino acids in here as possible, right? Any that are out there, we want to be very active in that absorption of it. So in this case, on the apical surface, we are going to use both primary and secondary active transporters. Right. So our primary, remember, are going to be things like pumps. Pumps directly use ATP. So an ATP binds to this and it uses the ATP of that binding to it to bring in that amino acid. We are also going to use our secondary active transporters, which of course are our co-transporters. And those co-transporters, as we know, are going to let something like sodium into the cell, something that really, really, really wants to come into the cell. We'll say, okay, sodium, you can come in, but when you come in, you got to bring that amino acid in with you. And remember, as we talked about in the last class, if we are going to use these co-transporters, then at some point we have to pump that sodium back out so that we can maintain that concentration gradient so it continues to want to do work. So we have all these ATP using processes, both directly and indirectly using ATP, because if there are amino acids in that lumen, we want to get them out of that lumen and into the cell. Of course, once we get those amino acids into the cell, they are stopped from being lost. And do we need to modify or do anything to them? No, again, a very silly question. I don't know why he keeps asking that. Maybe we'll find out. No, obviously we're not going to do anything with those amino acids. And if we're using so much energy to get them into the cell, do we really want to worry about using energy to get them out of the cell? No, we don't really want to do that. So primarily, we are going to use passive facilitated diffusion to get those amino acids out the basal surface, some type of carrier or something along those lines. All right. Once in the interstitial fluid, where does it go into? Which of our two types of capillaries, lymphatic capillary or blood capillary?
Blood.
Excellent. Enters a blood capillary. Let's make that a little bigger. Excellent. And once it's in that blood capillary, we talked about the pathway last time, so it should be even more easier to do this time. Yes, that's a phrase. So from that blood capillary, what does it feed into?
Intestinal vein.
Which feeds into?
Superior mesenteric vein.
Excellent. Which feeds into?
Hepatic portal vein.
Which feeds into?
Sinusoid capillary.
Excellent. Which feeds into?
Central vein.
Which feeds into?
Hepatic vein.
Which feeds into?
Inferior vena cava.
Which feeds into?
Right atrium of the heart.
Perfect. There you go. At least someone's got it down. Excellent. Glad to hear that. And there you go. Just that easily, we have identified the process and the pathway for our amino acids for protein absorption. Again, your book does a nice job of describing this. So we've done all of this. So again, the goal here, whether it's in the oral cavity with the chewing, the churning in the stomach, the pepsin here are enzymes of the small intestine from the pancreas, like trypsin, chymotrypsin, carboxypeptidase, things along those lines. And then those brush border enzymes, like dipeptidases and things along those lines, all help to break it down into those basic amino acids. Those amino acids cross the plasma membrane, primarily via co-transport and active transport. Notice, can there be some facilitated diffusion? Yes, but we are going to use primarily. So again, we're not going to stop it from coming in. Can there be some diffusion or facilitated diffusion? Yes, but what we really want to emphasize is that it is coming across the apical surface with energy. We are using as much energy as we can with both co-transporters and primary active transporters, using that active transport to get it in as efficiently as possible. Of course, once it's in, then we can get that facilitated diffusion out. And remember, we said mostly facilitated diffusion. There can be some code transportation to help to try to get it out as well. But the key is we are primarily using energy on the apical surface and primarily not using energy in the basal surface. No, as I said, you don't have to know the names of the brush border enzymes, just know them collectively as brush border enzymes. You do need to know the names of the common pancreatic enzymes that are produced. And I guess because proteins are so darn important, your book does a nice job of not just giving us this picture, this chart like we saw the last time, but they give us a pretty picture for this as well. Again, notice we have our co-transportation of that amino acid in through the apical surface, passes through the cell, and out that facilitated diffusion out that carrier on the other side and into a blood vessel from the interstitial fluid. So we have this pretty picture that kind of shows that path as well. Notice both these paths are pretty simple and straightforward. Questions on them? Look, this one even shows some pretty pancreatic enzymes and some brush border enzymes, all playing a role with it. It's all very simple, very straightforward, very fun. All right, questions on that? Well, if they were all the same pathway, then do you think I'd make you learn the same pathway?
Maybe not.
Probably not, absolutely. So that is a great question to ask. There are certainly some similarities in them, but especially between proteins and carbohydrates. But now we get to move on to lipids, and lipids are going to be an interesting case in and of themselves. So let's take a look and do lipids. Now I need to clear my board a little bit to be able to do this. Let's see how easily I can do this without messing things up too much. Perfect, that worked. Bye. Excellent. All righty. So our goal now is to talk about lipid absorption. Of course, as we know, lipids are broken down into what again?
Fatty acids and alcohol.
Fatty acids and alcohols. Again, primary alcohol is indeed glycerol, because again, remember our most common digestive lipid are triglycerides, but we can simplify by just saying alcohols. Excellent. Once again, we want to talk about the chemical breakdown and the mechanical breakdown of these. So let's talk about the chemical digestion. Oops. And I'll save you a little time because we know that there's the cardinal 3, and that is the mouth with mastication. We know that in the stomach, we get our churning. And in our small intestine, we get our segmentation.
You wrote chemical digestion.
Oh, you are correct. I did. Thank you for catching that. Excellent. Perfect. So there are the big 3. Those are the same for all of them. But let's ask the same question we asked for proteins. Is there any location or any type of additional special mechanical digestion that just takes place in association with lipids? And if so, where and how?
Bile emulsifies it.
Excellent, absolutely. And where does that occur?
Small intestine.
Excellent. In the small intestine, we have bile. And where's that bile produced from again?
Liver.
From the liver, excellent. Right? Or it could have been stored and concentrated in the gallbladder. Again, you probably don't have to write this, but let's be thorough. Bile from the liver, stored in the gallbladder, and its goal, as was mentioned, is that emulsification. That doesn't look, no, it's a U, isn't it? Emulsifies the fats. Takes big globs of fat and breaks them down into little ones, right? It's that hammer and that chisel. We're not breaking bonds. We're just breaking it down into little bitty pieces. Okay, excellent. What about chemical digestion?
Lingual lipase.
Okay, so we have that lingual lipase, which is of course made from the tongue. But remind me again, where is that active?
Stomach.
In the stomach. So the first place we start, and it's very limited, but we start the chemical breakdown in the stomach thanks to that lingual lipase that is produced from the tongue. Excellent. Where else? Excellent. Pancreatic lipase, of course, from the pancreas. And where is that active?
Small intestine.
Small intestine. Excellent. And we can't forget your friend and mine, also in the small intestine, the brush border enzymes. Excellent. All right, so here I need to cheat. Let's move this down a little bit. Move this down a little bit. And actually do that and that. All righty. Excellent. At this point, we have now broken down our lipids into fatty acids and alcohols. But remember, fatty acids and alcohols still don't really like water. So at this point, we have fatty acids and alcohols. And what happens is these fatty acids and alcohols mix with the bile salts. When they mix with the bile salts, they become these small structures known as micelles. Oops, that's supposed to be form. So what happens is we get these little green globs of the bile salts. And in the center of it, just for the heck of it, let's put a little yellow to remind us that there's some fat in here too. So these micelles are combinations of the fatty acids. And this is going to need to be smaller. Fatty acids. Alcohols, and the bile salts. All right. Now, all of these things for the most part are lipid-based or hydrophobic. As such, how are they going to get across the apical surface of the plasma membrane? Are they going to need ATP using transporters? Are we going to need channels? Are we going to need pores? Are we going to need carriers? Are we going to need any types of proteins?
Just diffusion.
Yeah, and not just any diffusion, but simple diffusion. Simple diffusion. No proteins, right? Nothing, no channels, no transporters, no anything. They are lipid soluble, so they're able to pass through the phospholipid bilayer of the cell without any problems at all. Complete simple diffusion into the cell.
May I ask you?
Yes.
I understand that bile salts, they like a coat for the fatty acids and alcohols, right? So
They're what?
like a coat.
Yes, sort of. Yes, they do kind of coat the outer surface in forming these micelles. Yes.
So why they need this coat if they just can simply diffuse into the cell? It's already broken into small pieces.
Yeah. I don't have a good answer for you for that. I don't know why it is that these things glom together. Yes, all of them individually are able to diffuse across the plasma membrane. So why they need to do it together, I don't have a good answer for that. In fact, what's even more interesting about that is what happens when it gets inside. My guess is maybe it stops the fatty acids and the alcohols from reassembling. That might be what it helps to do. It helps to keep them separate. Because notice what happens once they get in the cell. Once the micelle enters the cell, it breaks up. And once it enters the cell and breaks up, basically what happens is that they break up into the bile salts. The bile salts separate from the micelle. And those bile salts, as we already hinted at, are going to leave. As they leave, they're going to enter into the blood. Oops. Thanks. And remember, they get the blood capillary, as we know, goes back to the liver. And as we talked about, when it goes back to the liver, it can be recycled and reused. And it stimulates, well, I'm gonna move that on my mind. Goes back to the liver, stimulates the liver to make more bile salts, and it gets reused as a result of that. So as a result of that, and I guess I have to do that there like that. The bile salts enter into the blood capillary, go back to the liver, and get recycled again. What happens with the remaining components is the fatty acids and the alcohols reassemble into triglycerides. So they actually reform triglycerides. Then those triglycerides bind together with other lipids, like cholesterols, like steroid hormones, etc. All these other all these things, becoming this big, huge fat globule inside the cell. Now, that big, huge fat globule inside the cell isn't going to do anybody any good. So what happens is the outer surface of it gets coated with polar proteins. So we coat the outer surface with polar proteins.
Outer surface of the globe?
Yep, outer surface of these fats. So what happens is we get this big, huge chunk of fat inside the cell. All these fats, all these triglycerides and steroids and cholesterols and other lipids. And then what we do is then we take a big chunk of polar proteins and put them on the outer surface. And when we put these on the outer surface, right, we form one of those lipid protein, what we call a chylomicron. We form this big, huge structure called a chylomicron, which is a big chunk of fat that is coated in proteins that are polar proteins so that it can be dissolved in water. And now our goal is to get this out the basal surface. Now, if we're going to get this big, huge mass out the basal surface, can we have some type of channel, some hole in the plasma membrane that's going to make it easy to get something like that out?
No.
No. So how can we get large things out of the cell? It's a good way to get large things out of the cell.
Exocytosis?
Yeah, exocytosis, excellent. So we can use exocytosis. We can use vesicular transport to get it out of the cell, out the basal surface. They primarily leave via facilitated diffusion. So the bile primarily leaves via facilitated diffusion to go back into the blood and be recycled and reused. Now, two more things. Once this chylomicron is released from the cell via exocytosis, which type of capillary does it enter?
Lacteals.
Excellent. It is going to enter into a lacteal. And luckily, we learned about the lymphatic system, so we can trace the pathway back to the heart, starting from that lacteal in the small intestine. What would it feed that chylomicron? Where would it travel from that lacteal?
Oh, yes.
Where does it go from the lacteal? If only I had learned my lymphatic vessels. There we go. Okay, I like that. Let's do that first. So absolutely, it's going to go into the collecting vessels. And of course, that is going to be afferent and efferent. And remind me again, what happens with those afferent and efferent? What distinguishes them?
Euthyphro.
I agree. Afferent and efferent, what is the difference between, yeah, lymph nodes, absolutely. So collecting vessels, which will carry it through multiple lymph nodes. Absolutely. So we're going to have those collecting vessels, which is going to take it through those lymph nodes. So, so really all of them are towards the heart. Remember, this is all going back towards the heart. But you're also right, Ashley, in that these collecting vessels are ultimately going to feed into the intestinal trunk. And what does that intestinal trunk feed into?
Cisternoculi.
Yep, excellent, into the cisternochile. Excellent, which feeds into the thoracic duct. Excellent, which feeds into the
Left subclavian vein.
which feeds into the
Left brachiocephalic vein.
which feeds into the
Superior vena cava.
which feeds into the,
Right atrium.
there we go. And we've made it back to the heart. Questions on that? All right, excellent. Let's take a look at the pretty pictures from your textbook that do an almost perfect job of showing this as well. Let's see. So again, here we see those examples of how it's broken down. Lingual lipase is released in the oral cavity, becomes active in the stomach. Our bile salts chemically, mechanically, our pancreatic lipases chemically break it down. And again, those triglycerides are broken down into fatty acids and alcohols. They mix with the bile salts to form those micelles, and the micelles enter the apical membrane just via diffusion. Simple diffusion. No proteins are needed because they're lipid soluble. Once inside, those fatty acids and alcohols are going to form back into triglycerides. They're then going to be added to other lipids like steroids, cholesterols, phospholipids, other things like that, and coated with polar proteins to form those chylomicrons. A chylomicron is a big structure. So the only way out the basal surface is via exocytosis, where it goes into that lacteal and travels on that merry path back to the heart. Now, again, your book's got these pretty flowcharts on that nice table. And again, they have this great picture. And I love this picture, except they totally screw up the size of these things. I understand why they're trying to do this from a perspective standpoint. Oh, great question. So, okay, let me answer that as soon as I finish this. So notice we have that big, huge fat globule. The bile salts emulsify it, break that one globule into a bunch of small ones, allowing the enzymes to get in here and break them up. And as the enzymes break it up, they mix with the bile salts and they form the micelles. The micelles enter the apical surface by simple diffusion, and then they are mixed with other lipids. So I mentioned like steroids, I mentioned cholesterol. Remember, there are two different types of cholesterol. There's the cholesterol can be a positive and healthy thing in helping to make plasma membrane. It helps to give more structure and integrity to the plasma membrane. So not all cholesterol is necessarily bad. And again, when I say that they are being added together, I don't mean that we're making these huge complex compounds that are made up of cholesterols and triglycerides and all those things. What I'm saying is I'm grabbing a bundle of random things and bundling them together. So some of those things will be triglycerides. Some of those things will be phospholipids. Some of those things will be, steroids or cholesterols or lipid soluble vitamins, things along those lines. We're just taking a bunch of stuff together and putting it in a box so that it can be transported. So we're not forming big, huge, complex molecules. We're just coating all of these things together so that they can be shipped. We're packing them into a box. It's like a care package that is going to be sent out. All right. So, hold on. So, like I said, I love this picture. I think they do a great job of showing what happens. Then it forms that chylomicron and enters the lacteal. However, the one point I want to emphasize from this picture is which is bigger, a micelle or a chylomicron?
Chylomicron should be bigger.
Yeah, chylomicron should absolutely be bigger. I think the problem they got into is a problem of perspective. They showed these massive fat droplets, and to make them in perspective to size of the cell, right, it would have been far more than the page. So they ended up shrinking the perspective down. And so because of that, we have this weird view where the micelles are larger than the chylomicrons. But if you ignore the size of everything, then you can clearly, it does do a nice job of showing that path. So that's my only big major complaint about this picture, but I think it does a nice job of doing that. All right, questions on that?
I have a question.
Yes.
As you can see in the step 3, it says that monoglycerides and fatty acids leave micelles before they diffuse this into the cell.
Well, I know I wouldn't say. I appreciate that it's saying that, but remember the bile salts have to enter as well. These are all entering together.
Okay.
Okay.
But you can see that, right? I understand correctly.
Yeah, I do understand, but I think they're just trying to simplify it here. They're focusing on the fats, but you have to remember that the bile salts are entering in as well. The bile salts are being recycled as well, and it is the same mechanism by which they do that.
Okay, thank you.
Yep. All right. So that is 3, but I did promise a 4th possible pathway that you might have to know, right? And the obvious thing it would be the nucleic acids, right? Breaking them down, how we would break them down into nucleotides. However, if you remember, that really only happens in one place, in the small intestine, with the deoxyribonuclease and the ribonuclease that is released by the pancreas. the brush border enzymes, the pathway across the apical surface is basically the same as proteins. The basal surface is the same as proteins. The pathway from the interstitial fluid is the same as proteins. So it doesn't make a lot of sense. It doesn't make a good essay question. It doesn't make a good option. Instead, the fourth pathway is going to be something else really important that needs to be absorbed. And what's that other really important thing that we need to be absorbing from our digestive system?
Nucleic acid.
Water, right? Because again, I said nucleic acids are important, but it's a boring pathway, so it's not worth asking. The fourth pathway is the absorption of water, right? Again, we here in Sacramento appreciate it better than most. How much water you absorb from your digestive system, how much fluid you put into your digestive system has a dramatic impact on the function of your body. All right. Over 9 liters of fluid. And again, depending on the number of times you eat, the volume of the food you eat, the composition of the food you eat, this will vary. But on average, right, 9 liters of fluid enter our alimentary canal in a 24-hour period of time. And only a small amount of that is from the food and drink that we ingest. Right? As we went through all of our different accessory structures and glands, and we talked about 1 liter of saliva and 2 liters of gastric juice and 1 liter in bile and 2 liters of pancreatic juice and so on and so forth that's going through here, right? Notice that all of these are, you know, close to 7 liters of fluid that is coming out of our body. Do you have 7 liters of excess fluid in your body you can afford to release?
No.
Do you want to be defecating 7 liters of fluid, right, outside of your body in a 24-hour period of time? Doesn't sound like a whole heck of a lot of fun. Absolutely not. So one of the most major things we need to do is not just absorb the water from what we eat and drink, but also absorb the water from all of those secretions that we are releasing into the alimentary canal as well. Notice, and I think I highlighted this previously, the vast majority of that reabsorption is occurring in the small intestine. You know, I think that there's this misnomer that the large intestine is the primary location where water reabsorption occurs. And there is reabsorption that occurs in the large intestine. I think the problem is that there's not a lot of other stuff that happens in the large intestine. So probably the most important thing that the large intestine does is absorb water, because not only is it one of the few primary functions it has, but we are poignantly aware of when there are problems with it. If it doesn't absorb enough water, what ends up happening? If your large intestine does not absorb enough water, there you go, exactly. It's anatomy and physiology class. You're allowed to say diarrhea, right? You'll have more water in your stool, and that can lead to diarrhea. If your large intestine absorbs too much water, what happens then? Yeah, your stool could become compacted and constipation can occur as a result of that. Absolutely. So we are very poignantly aware of how important water absorption is in the large intestine. But as you can see, clearly the majority of the absorption of water does indeed occur in the small intestine. Yes, so exactly. So not, well, you can think of it either ways. The fecal matter goes through your large intestine too quickly, so you're not able to absorb as much water from it. Or if there's some type of problem where you're not able to pull as much water out of it, then that's what causes diarrhea. But yes, often what happens is some type of irritation, something along those lines does cause the fecal matter to move through too quickly, not giving the large intestine enough time to absorb it. And so yes, then you get a very runny stool or diarrhea as a result of it. Excellent. So there you go. So we have those primarily absorbed via osmosis, which again, remember, is just diffusion of water. And once inside, it is going to enter both the vascular and the lymphatic capillaries. So while obviously there's no chemical and mechanical breakdown of of water, you want to say where the water is coming from. You want to say where most of the water is going. And lucky you, since we want to get back to the heart and it takes two routes, you get to describe both the the blood vessel capillaries and lymphatic capillary pathway back to the heart when describing the reabsorption of water.
Describe both?
Yep. Hey, if you don't have to talk about chemical and mechanical breakdown, I got to do something to bulk up the question, right? So there's two pathways that water would take to get back to the heart, and so you would need to describe both pathways.
Makes sense.
Yep. All righty. The goal, notice about 9.3 liters enter into the alimentary canal, about 9.2 liters are reabsorbed. So on average, only about 100 milliliters of water should remain in the feces during that 24-hour period of time. Enough to maintain the integrity of it, but enough to keep it lubricated so that it moves smoothly through the large intestine. And with that, we are done with the small intestine, and we can move on to the large intestine. All right, questions on that? All righty, that is our absorptive pathways. That is our finish of our discussion of the small intestine, but we're not quite done with our merry way through the alimentary canal. So let's go ahead. This is a good spot to take our first break. Let's take our first break. It looks like it's 12:55, so we will take a 15-minute break, come back at 1:10, and at 1:10, oh, it's this. Yep, so yes, in the case of the reabsorption of water, it is just water, H2O, absolutely. So we're not really absorbing other things, are there? So okay, great question. Maybe I went through this too fast, so thank you for asking this. All right, so let's do this. Right? If we think in terms of water, there really is no mechanical digestion of water that is taking place, right? Water is just water. There is no chemical breakdown of water. Water is just water. So water in its form just needs to pass through the apical surface. And it is going to pass through the apical surface because it is water. And we'll just put it as a blue circle here. It just passes through. Be a simple diffusion. Right? Simple diffusion. Water gets through the plasma membrane by simple diffusion. Of course, being, anatomists and physiologists and all the fun things that go along with that, they like to give a special name to the simple diffusion of water. And what is that simple name that they give to the name that they give to the simple diffusion of water?
Osmosis.
Osmosis, absolutely. All osmosis is the simple diffusion of water. Water moves down its concentration gradient from a high concentration of water to a low concentration of water. It goes to where there's more stuff. And if you think about it, we just talked about all this active and passive movement of stuff across, oops, yeah, where am I writing this? Okay, try that again. Lots of active, oops, and passive movement of stuff like amino acids, like simple sugars, like fatty acids, like all those things, right? All of this generic stuff into the cell. And so not surprisingly, water follows. Simple diffusion, water follows all of that stuff because water follows stuff. Water follows salts into the cell via simple diffusion. course, once that water gets in the cell, it needs to get out the cell. And as we just talked about, there's plenty of movement of materials outside the cell. Water is going to follow that. And once again, it's going to leave via osmosis. Follows the stuff. So notice getting the water into and out of the cell is a really simple process. Most of it happens in the small intestine. And we're able to get the water back here into the interstitial fluid. So again, very simple to get the water into the interstitial fluid. However, once it's into the interstitial fluid, how many paths can it take to get to the heart?
Yeah.
2. Absolutely. Water will both enter the blood capillary.
He gave him examples, but he didn't explain it. Huh. Damn.
I'm sorry, what?
He never explained it.
What didn't I explain? What can I help with? Okay. So it's going to enter both the blood capillary and the lacteal. And so if it enters both the blood capillary and the lacteal, then you would have to describe both pathways. To the heart, because it will take both pathways to the heart. So does that make more sense? What I'd be looking for water. And really, again, notice it's really more water reabsorption, because while, if you think about it, of the 9 liters of stuff that we're reabsorbing, close to 7 of it is stuff that started in our body and we released. So if you called it water absorption, that would be fine, but it's really probably more appropriate to call it water reabsorption. All right, so I apologize if I rushed through that and that didn't make sense, but hopefully it makes sense now. Questions on this?
I have a question.
Yep, I have answers.
If not taking the food, like fasting, we lose weight because of losing water?
Yes. So water weight is one of the faster ways to lose weight. Again, if you think about it, 60% of our body weight is water. So whether, and again, when I always think of it, I always think of back to high school. In high school, I wrestled, and luckily I happened to be very close to the weight that I wrestled at. So monitoring my weight wasn't as big of an issue. However, if you were one of these people who had to make sure that they lost 10 pounds to be able to make their weight class, they would be practicing in class wearing plastic bags. So they would wrestle wearing plastic bags in class. When they were on the bus, they'd be sucking on a hard candy to produce saliva and be spitting that out, right? In college, I didn't see any of this in high school, but in college, people will take diuretics to flush water out of their system. So yes, a lot of the ways that we can, if you wanted to rapidly try to lose weight, right, for something like a wrestling thing or something along those lines, they can do that. Now, that isn't a healthy way to lose weight rapidly, but it can help you to lose weight rapidly that way. So yes, some of the early, when any weight loss program you're in, much of the early gain is from the loss of weight and not so much the loss of adipose or anything else along those lines. Yes.
So in this case, water is not reabsorbed because it's...
Well, no, you're still reabsorbing water, but what they're doing is they're losing water other ways, right? So for instance, if you take a diuretic, that tells the kidneys to stop reabsorbing water and you lose more water in your urine. If you're producing sweat and evaporating that sweat away or producing saliva. I guess that would be the one example, saliva. If they're spitting that saliva out instead of swallowing it, then less fluid is entering into their alimentary canal that way. So it's just another way of losing water. But typically, as far as I know, there aren't any weight loss mechanisms where you can reduce how much water your digestive system absorbs. And as we mentioned, I don't think that's something you would necessarily want to do, because if your digestive system doesn't absorb that water, then that means it has to come out the other end.
Okay, thank you.
Yep. All righty. Questions on that? So like I said, one of these 4 pathways, I guarantee will be an essay question on the exam. And the fun thing about doing this online is I have no idea which one. Each one of you is going to get a different one. So it will be random. But I guarantee they will be on there. So make sure you understand this material. All right. Questions on that? All right, with that, we are done with our small intestine. With that, we are done with our absorptive pathways. So as I mentioned, let's go ahead and take our first break. It is now 1:02, so let's go ahead and take a 15-minute break, come back at 1:17. And at 1:17, we will restart. All righty. Any questions? All right, see you guys in 15 minutes. Oops, didn't mean to do that. Wanted to do that. Excellent. All right. Any questions before we dive back in? All right, perfect. Then let's go ahead and get started. We are going to talk now about the last major organ of our alimentary canal, and that is the large intestine. It is divided up into 3 distinct regions. Those regions, where's my highlighter, is the large pouch-like cecum at the proximal end, the large pouch-like rectum at the distal end, and pretty much everything else in between, which is commonly referred to as the colon. However, this whole thing commonly in between, known as the colon, is divided up into its own distinct regions. We have the part of the colon that goes up the abdominal pelvic cavity. So of course is the ascending colon. We have the part of the colon that goes down in the abdominal pelvic cavity, which is the descending colon. We have the part of the colon that goes across the abdominal pelvic cavity, that is the transverse colon. And we have the 2 curvatures to it. We have this first curvature here, which is the right colic flexure, because it is the bend in the colon on the right side of the body, but it happens to be next to the liver. So it is also sometimes referred to as the hepatic flexure, which is an acceptable term. We then, of course, have the left colic flexure, which happens to be next to the spleen, so is also called the splenic flexure. And then lastly, we have the S-shaped region of it that is given the name sigmoid colon. So those are all the specific regions of the colon. Remember also, as we talked about, and we'll emphasize this with color, the transverse colon is intraperitoneal. So it has a little bit of mesentery that helps to anchor it in place. And what do we call that mesentery that anchors the transverse colon in place?
Transverse. Amen.
Transverse mesocolon. Excellent. Our sigmoid colon is also intraperitoneal, so has a little bit of serous membrane that anchors it in place. And of course, it is called the sigmoid mesocolon. And remember that our Ascending and descending colons, of those portions are retroperitoneal. So they help to anchor against the back wall and are anchored in place that way. So again, this whole thing that we call the large intestine can be divided up into basically 8 distinct parts. Now, like every organ we have seen in the alimentary canal, at least up to the rectum, right? So basically everything up to the rectum has the same basic anatomy as the rest of the alimentary canal from the inferior esophagus down, where we have a simple columnar epithelial tissue that is part of a mucosa, submucosa, muscularis externa, and either an adventitia or serosa, and notice this one gets both. But it also has some specializations to it as well. One of those specializations, as you look at the colon, are these pouch-like collections of the colon. The large colon, the large intestine has this excess muscle tension in it, which causes this pooching or pouching of the large intestine. And this distinct structure, this pouch-like structure right here, is what is known as a haustrum, singular, or the plural of them are the haustra. Now, one of the reasons these haustra form is as you look at this, and this illustration does a nice job of showing 2 examples of it, but there are some enlargements, and let's change colors again to, I think this pink will show up. There are these enlarged bands of the longitudinal muscle layer. So obviously we have a circular muscle, we have a longitudinal muscle layer of our muscularis externa. That is the same. We have seen how the circular layer can enlarge to become sphincters. Well, in this case, we have bands of the longitudinal muscles that enlarge and help to keep extra tension along the length. And that's where this pouching and pooching of the large intestine comes from. And these enlargements of the longitudinal layer are what are known as the tenia coli. So there are several, again, our picture here shows 2, but there's several enlargements of the banding of that longitudinal muscle, keeping excess tension in our large intestine to cause that pouching. Way back in the lymphatic system, we talked about this worm-like extension that comes off of the cecum, known as the vermiform appendix. And what did we say that vermiform appendix did again?
Bacteria?
Yeah, we talked about it, how it's a thin fluid-filled tube that can house some of the seed colony associated with our bacterial flora and fauna of our large intestine. Of course, it is lymphatic tissue, which can become inflamed and enlarged and cause that condition that we call appendicitis, which can be a bad thing, but it is a functional portion of the lymphatic system. Lastly, as you notice, and again, there's a couple things here that I want to point out. Notice also, and here I'll use yet a different color. Notice here we have the cut edge of the greater omentum. So remember, the greater omentum is that big, huge drape-like mesentery that is going to drape over the top of the small and large intestine, comes off of the greater curvature of the stomach, drapes down, and then comes back up and connects to the anterior side of the transverse, of the transverse colon. I make a point of emphasizing this because, as you can see, there is some adipose that is not unexpected within that mesentery. So when I talk about the epiploic appendages, I am not talking about the greater omentum. As you can see here, along the outer wall of the large intestine, there are these fatty appendages that are kind of attached to and anchored to the large intestine. Their functions are not fully understood. It is stored adipose, so again, it's stored nutrients. It may help to play a role in providing easily accessible nutrients for the bacteria to maintain the bacterial colony because we eat intermittently but the bacteria's life cycle is such that they are much more continuously metabolically active, so they may help to play a role in that, but their full function is not completely understood. But a consistent characteristic we see along the colon are these fatty appendages known as the epiploic appendages.
Sorry, you cut out for me when you said what the tinea coli is. What was that again?
The tinea coli are enlargements of the bands of the longitudinal muscle, keeping extra tension in the large intestine so that it's shorter. And as it shortens, that's what causes the pouching of the haustra.
Okay, thank you.
Yep. Excellent. All right. Questions on those? So here is some of the gross anatomy associated with the large intestine. Of course, we need to be able to recognize the histological microscopic anatomy of this. And as we look here, for instance, at a histology slide, we can see some of the key characteristics that are going to help us to distinguish it. For starters, we see our 4 layers. We see a mucosa, we see a submucosa, we see a muscularis, and we see a serosa or an adventitia on the outer surface. Notice how many muscle layers do we see here?
2.
2. Which muscle layer is this?
Circular.
Circular. What muscle layer is this?
Should be longitudinal.
There you go, exactly. What is the plane of section of this layer of smooth muscle? Transverse, cross section, absolutely, I would accept that. And what is the plane of section of this layer?
Sagittal.
Okay, sagittal is not bad, but really in simplicity, it's just longitudinal. They call that a longitudinal section, right? So a cross section is when you cut across the body of a cell or a wall layer in this case. And a longitudinal is when you run along the length of it. So yeah, I would accept sagittal for that, but longitudinal is probably a better description. So yeah, so notice we can tell which layer is which, not by the plane of section, but by its relationship to the lumen. Yep, absolutely. So here, let's use the highlighter so we can see it more clearly. These bands of muscle right here is the circular layer. So all of this, not just what I highlighted, but all of this is the circular layer. And all of this is the longitudinal layer. And notice here, we can see the cells in parallel, whereas here, we just see the cells in cross-section. So again, we can clearly see that these are 2 distinct planes of muscle layer because of the differences in the orientation of the 2 layers. Here, the cells are all longitudinal laid together. Here, we just see the cross sections through them. So the 2 layers are very distinctly different. Go ahead.
If you cut it differently, like vice versa.
Yeah,
So-
absolutely. If instead we had cut this tissue this way, then yes, you would see the circular layer in a longitudinal section, and you would see the the longitudinal layer in a transverse section. Okay. Yes and no. We can see out here that this is the protective layer on the outer surface of it. But remember, the problem is we're here in the large intestine. So can we tell with 100% certainty whether this is a serosa or an adventitia? No. So we would use the term interchangeably, and I would accept the term interchangeably. If I pointed an arrow at here and asked you to identify this specific sublayer, or the specific layer, then I would accept either serosa or adventitia. The longitudinal layer is always furthest away from the lumen. Yes. Yeah, the circular layer is always the one that is closer to the lumen. I want to make one other point. So more questions. I love these questions. As I've said many, many times, the only stupid question is the question not asked. So again, if this is not making sense, then please, by all means, ask these questions, because I love this. Because again, Heck, you guys are paying good money for this material. You need to make sure to take this class, you need to make sure you know it and understand it. I made a point of starting with the muscularis externa layer because if I look up here at the mucosa, notice I don't see villi, so I know I'm not in the small intestine, but I do see some fairly large glands. And these fairly large glands have a lot of goblet cells in them. Notice it is somewhat similar to the anatomy that we saw in the pyloric region of the stomach. Now, the pyloric region of the stomach would have a much thicker mucosa, but again, that's a little bit subjective. So if this mucosa looks so much like the stomach, like the pyloric region of the stomach, and of course, here's that muscularis mucosa. If this one looks so similar to the stomach, how can I be with 100% certainty knowing that I'm looking at the large intestine and not the pyloric region of the stomach?
Stomach has 3 layers of muscularis.
Exactly. While the mucosa may be trying to trick me that I'm looking at the stomach, the fact that I only have 2 muscle layers here is a dead giveaway. I'm not looking at the stomach. Oblique. Remember, the 3rd layer is oblique. So oblique, circular, and longitudinal. Okay, excellent. But what we do have here, no worries, what we do have here are the intestinal glands. The intestinal glands of the large intestine primarily make mucus. Really, the primary secretion of the large intestine is mucus. So we have all these goblet cells making the mucin and releasing them from these intestinal glands. And so that's what we're doing. We see these intestinal glands, we see these intestinal crypts, which is what they're called here in the mucosa. And so here, like I said, because of those crypts, because of those goblet cells, it may trick you into thinking it's the pyloric region of the stomach, because there was a lot of goblet cells production in there. But like I said, the fact that we can see the mucosa, the muscularis, and see that there is just 2 layers to it tells us that this is the large intestine. All right. Questions on that?
What are in the submucosa? It's a glands.
Oh, no, actually, great question. This is not something that is necessarily indicative. It just happened to be on this particular slide, and I didn't think it was worried enough to take one that isn't. Anybody have any idea what these might be? What this and this are?
Lymphatic follicles? No.
Not a bad guess. It does look a bit like that, but look more closely. Look at this structure and this structure that are similar structures, but not the same structure. Bacteria is not a bad guess, but we wouldn't necessarily want that in the submucosa. Think about what kind of things did we say we would find in our submucosa? Big glands and
Lymphatic nodes.
some lymphatic vessels, but those aren't, not lymphatic vessels, lymphatic nodules, but these aren't lymphatic nodules. What else would you find? There you go. BVs. What are BVs?
Blood vessels.
Blood vessels. And there are 2 of them here, 1 and 2. What type of blood vessel is 1?
Vein.
Yep. And what type of blood vessel is 2?
Artery.
Artery. Notice the artery has a thicker muscular wall to it. The vein has a larger lumen. So we actually have 2 blood vessels in here. So again, as we know, the submucosa can contain large blood vessels. And here we see a nice example of that.
What about that other structure to the left?
This is another vein.
Okay.
Yep. These are more in cross section. This is more of a longitudinal section. But again, we can see that simple squamous epithelium, and all these red things that we're seeing are red blood cells. These are the red blood cells inside of these blood vessels, the veins and the artery. Yep, one on the left is a vein. That is correct. All right, excellent. Any other questions on the histology? I think we got everything off the histology list for that one. So yes, so the crypts of Lieberkühn are the intestinal crypts. If you like to, again, Lieberkühn is a great term to say. Good old Bob Lieberkühn is the one who described these. That is that term for that. But just intestinal crypts is fine as well. You can just say intestinal crypts. You don't have to say Lieberkühn, but Lieberkühn is fun. I will give you that. I wouldn't say that they house phagocytes in them. Like all mucous membranes are going to have some protective cells and things like that along them, but no, not necessarily in the intestine. In fact, we probably wouldn't want our own phagocytes in our large intestine, because what is our large intestine filled with?
Bacteria.
Yeah.
Bacteria, absolutely. So we wouldn't necessarily want our phagocytes running rampant inside of the lumen of our large intestine, because they could potentially stimulate an immune response to the things that we want to be naturally living there. Excellent. All right, so that is the anatomy of the large intestine. Let's talk some physiology for it then. And starting first with its mechanical movements, because we did say there were some special movements associated with them. Again, obviously, as we talked about, that gastroileal reflex started with the stretch of the stomach, produced that gastrin. So remember, once again, at that gastrin, there it is. Gastrin is going to stimulate the relaxation of that ileocecal valve, or the ileocecal sphincter, the ileocecal valve. And that is, the job of that is to, again, release the chyme from the small intestine into the large intestine to make room for more food. So we talked about that as well. But there are some other movements that are unique to the large intestine. One of them are haustral contractions. So if you think about it, we have, and I can draw it here, these pouch-like structures. So we'll throw 3 haustra here together. And what will happen is that just one of these pouch-like haustrum will contract. And as it contracts, it does a couple of things. It is going to mix and compact the chyme. At the beginning of this process, it is still chyme, but eventually, as it is processed, it becomes feces. So this chyme/feces at this point within this one particular haustra, mixing it with the bacteria, helping to extract water and other materials from it. And of course, if we're increasing the pressure in here, that is going to cause the chyme to leave this haustra to the one next to it. However, it's not always going to go forward. It can go backwards in this process as well. So when this one haustra contracts, the chyme will move to the 2 other ones adjacent to it. And then one of those will contract and it'll move back and forth. So as you can see, this is very similar to segmentation. There is some motility, there is some movement occurring, but it's much more about mixing and compacting that chyme, mixing it with the bacteria, extracting the water and the nutrients, compacting it so that it can be released as feces, and those types of activities. So it isn't a true propulsion, right? However, there is going to be propulsive movements. While it can undergo minor peristalsis, the major movement in the large intestine occurs thanks to these movements called mass movements. Again, these mass movements occur on average 3 to 4 times a day, but again, it is directly related to how often you eat, the volume of food you eat, the composition of the food you eat, because these are stimulated by gastrin. But what happens is we get these long full peristaltic contractions that spread throughout the entire colon. So through all those haustra, with a goal of ultimately moving at the end of the process, feces into the rectum for removal from the body. That same gastrin that causes the opening of the ileocecal valve also increases the motility. So like I said, that gastrin is going to increase the motility of the large intestine. And again, I use the example that we talked about before. Little kid sits down at a table, has 3 bites of food, and then has to run to the bathroom and poop. Because as his stomach stretched and released gastrin, it opened the ileocecal valve, it started the movement in the large intestine. They have a small large intestine, so there's not a lot of place to go. They have a small rectum, so not a lot of place to go. Stretch of the rectum, and suddenly they need to urgently go and poop. All right. Dogs too. Okay, there you go. Yeah, there's smaller sized dogs, I imagine, would be like that as well. Excellent. Questions on that? All right. Let's talk about the chemical activity in the large intestine. As I already mentioned, the only major secretion that is produced by the large intestine is mucus. Now, again, notice I say little to no enzymatic activity that occurs. What I mean by that is driven by the large intestine, right? There is no pancreas-like gland releasing enzymes into the stomach, no salivary, no lingual glands, no gastric glands producing enzymes and releasing those enzymes into the large intestine. The only major substance that the large intestine produces is mucus, right? But that doesn't mean that there isn't enzymatic activity taking place there. It's just it's the bacteria that are doing the work. It's not the large intestine that's making the enzymes. It's not the large intestine that is doing the work. Instead, your digestive system has this major flora and fauna to it, bacterial flora and fauna, that is elaborately involved in the digestion of certain components. And as we talked about, I think I'm pretty sure we already talked about this before in this class, they're finding more and more that the composition of the bacteria in your large intestine can have major impacts on autoimmune disorders and allergies and all sorts of other, what's the term I'm looking for? Mental conditioning, all sorts of types of things that it may play an important role in. And we talked, I think, about this class about fecal transplants, where if someone doesn't have the right bacterial composition, right, they can actually get a suppository of somebody else's feces that has the right combination of bacteria, and they see dramatic improvements in many, many ways. Again, these studies are still relatively small, somewhat anecdotal, but it is a huge area. You say, oh my, did we not talk about this in this class? I thought we had talked about this in this class. Yeah, they're finding that the bacteria in our large intestine, while obviously, yeah, I thought we did. While obviously playing a major role in the breakdown in some of the major components that we need to absorb through our large intestine. We will talk a little bit about those in a second. But like I said, they're finding that it plays a much bigger role in things like triggering autoimmune disorders or lessening the symptoms associated with autoimmune disorders. They believe some types of food allergies may be associated with the bacteria that are located in there. And like I said, there's at least one study that I know of that looked at differences in cognitive abilities associated with differences in the bacterial components. So there's something about that bacteria in there that may influence your mood or your behavior or your cognitive skills. I mean, it's an incredibly interesting field of study. One I would not necessarily want to have anything to do with myself, but it is an incredibly interesting field of study where a tremendous amount of work goes into. And like it or not, as a father of a daughter with major allergies, it is something that is on my radar. All right. Yeah, exactly. I'm used to the other end. I'm a neuroscientist by trade, so I don't deal with bacteria. Bacteria don't have brains. So if you don't have a brain, I'm not interested in you. So I don't like broccoli either. Broccoli and bacteria. Can't stand either of them because they don't have brains. All right. So while the large intestine doesn't have any major functions from a secretory standpoint, doesn't mean there isn't some important things that go on here. Again, we did talk about the importance in its role in absorbing water. This is one of the locations where additional bile salts can be reabsorbed in here. Other different types of organic materials can be here. And probably one of the most important thing our large intestine does, it is the site of the absorption of vitamin B12. B12 typically is not released from the food we ingest until the bacteria is able to break it down. So bacterial activity releases B12 into the lumen of our large intestine. And what did we say we needed to be able to absorb that B12 into our body from the large intestine?
Making erythrocytes,
I'm sorry?
making blood.
Oh, no, you're correct. That is what we need the vitamin B12 for. Absolutely. We need vitamin B12 to make red blood cells. It is vitally important. However, if I want to absorb vitamin B12 from my digestive system, there you go. What I need is that intrinsic factor. So only in the presence of intrinsic factor, oops, intrinsic factor, cannot talk and type today to save my life. Intrinsic factor, am I able to absorb B12 in the large intestine? And someone remind me again, what makes that intrinsic factor? Parietal cells, excellent. Remember, as we talked about, that intrinsic factor from the parietal cells, this absorption of the B12 that intrinsic factor allows us to do is really the only vital function of the stomach. Is the stomach super important in helping the digestive function, acting as a storage location, acting as a place to start the chemical and mechanical breakdown, in distributing the chyme in a slow and controlled manner into the small intestine? Absolutely. You definitely want to have a stomach rather than not have a stomach. But while all those things help, as we talked about, you can definitely survive without a stomach, except for this. This is the one thing that your stomach does, that production of that intrinsic factor, allowing us to absorb B12, is the one thing that it does that is vital for human existence. We could not survive without. And then, of course, the other important function of the large intestine is to process that chyme into its last iteration, its last form, and that is feces. And then storing, compacting, preparing that feces, and ultimately then removing the feces from our body. I want to talk a little bit about what the bacteria is doing in your large intestine. But again, this is one of those times where I know when you get into microbiology, you guys will talk about this in a lot more depth, so I don't have to go too far into it, which is good because, as I said, bacteria don't have brains, so I don't like them. Bacteria, jobs is to break down the portions of the food that our enzymes are not capable of breaking down themselves. And that allows us to release some nutrients from those foods that we otherwise wouldn't be able to obtain. Some of those are things like vitamin K and vitamin B12. There are other electrolytes that can be released and other lipid-soluble vitamins that are released by that bacterial activity. Of course, we do pay a bit of a price for this bacterial activity. Bacteria primarily break these things down anaerobically, right, without oxygen, right, or what we call bacterial fermentation. As a result of that breakdown process, there are many undigestible carbohydrates that are converted into carbon dioxide and methane gas. This produces that flatus, right? That gas that we pass as a result of that. the bacterial breakdown of certain proteins or ammonias or indosols or hydrogen or those types of things produce hydrogen sulfide gases or ammonias. And these are what give the odor to that flatus. And as we also talked about, that bilirubin is broken down into stercobilin and urobilin. Remember, the urobilin is reabsorbed. And the stercobilin stays in the fecal matter, giving it its color. For a while in history, there was an interest in studying the composition of feces, and it was believed that a lot could be told about the health of an individual by the color, the weight, the size, the consistency of the feces. So many medieval doctors would collect the feces from the kings and queens that they were looking for, and they would be able to discern all sorts of information and important things about them from that. But really, what it tells you more about is the condition of the bacteria inside of you as well. All right. So like I said, there's far, far more we could go into this, but we can easily, like I said, you'll get a lot of this in micro. Random question. I like it. Excellent. Go ahead, Alex. My answer is blue. I will be honest. I have never heard of that before. Homeless people or anybody else huffing their own fecal matter. And I'm not sure why huffing ammonia would, are they supposed to get some kind of high or something off of that? I will be honest, I have not heard of that before. Yeah, it doesn't necessarily sound like a pleasant experience. I- um, okay, so yes, fecal matter contains ammonia from our bacterial decomposition of proteins. That is correct. I am not certain what inhaling ammonia would, how that would affect you. I'm not sure. As far as I'm aware, I mean, again, when you say something like huffing, obviously we're thinking of something to try to get into some type of altered state of consciousness, like getting a cheap way of getting drunk or getting high, or somehow getting into some other altered state. I am not aware of ammonia having that type of cognitive effect. That doesn't mean that it doesn't. It's just not something that I'm aware of. But I guess that is true. Yeah, Ryan. Actually, that's a good point, Ryan. If you right, when someone, if if an athlete or something like that, a football player gets hit in the head, and one of those things they do is they break the ammonia underneath the nose to try to help to wake them up and make them more alert. I think if I was homeless on the side of the road, I think being alert is probably the last thing I want. And I guess unless you want to make sure nobody's shivving you or taking your stuff or something like that, but pinch yourself or something. There's got to be better ways to stay awake. Well, but okay, there are mushrooms that can be grown, and it's not including psychedelic mushrooms that are grown from cow poop, but that's just because it's a very potent fertilizer. It isn't necessarily that the mushroom is growing out of the poop. It's used, the seeds from the mushroom are planted in the poop, and the poop is acting as a fertilizer for it to grow. Like I said, I'm not familiar with it. Like I said, if I was homeless, I'm not sure I would want to be that alert. I think there's maybe an advantage to being incoherent, but I haven't, I can't think of a good example of why you would do that. And like I said, I think Ryan reminded me that, yeah, most of the time that I think of sniffing ammonia, it's to help to alert someone, make them feel more awake. I guess. I mean, we haven't gotten to the urinary system yet, but obviously one of the things we'll talk about in the urinary system is there are some homeopathic beliefs about urine, especially like the first urine of the morning, bathing in it, drinking it, washing your face with it, soaking your fingers in it, that there's somehow some benefit to that. So are there some theories that there might be some benefit to feces? I guess I'm not as familiar with those, but if we've learned anything this year is people will believe anything. So who knows what when someone's told someone to believe. I don't have a good answer for that. That's awesome. I had not heard of that before. Yeah, and he also has a tattoo on his face. So I'm not sure I'm going to take what he says for gospel either. All right, excellent. You weren't kidding. That was a random question. That's awesome. All right, excellent. So again, wait, the Tyson thing is called Jemkin, or the sniffing the feces thing is called Jemkin? All right, I'll have to look that up during the next break. I haven't heard of that. Hold on, I got to write that down. I have a new essay question for us now. Describe Jemkin and why people do it. Well, I gotta make sure I spelled that correctly. I'm kidding. It's not going to be an essay question, but I am always interested. Oh, Jen Kim. All right, I will have to look that up. That's hilarious. Yes, so especially things like a tapeworm or something along those lines, absolutely. The buds of those tapeworms can be found in feces. Defecation that occurs at night when things like hookworms are more active, those can actually be present in the feces as well. So yes, and again, that a lot of times they do that for dogs as well. If you have something wrong with your dog, you have to take a feces sample into the vet to be able to help to diagnose that as well. All right, so thankfully for the last time, it varies based on the volume of food, the number of times you eat, the composition of the food you eat, but on average, it takes about 12 to 24 hours to completely break down and absorb all of the materials that we need from that chyme. And finally, after that period of time, that chyme has been processed to its final iteration, feces, right? So we went from food to bolus to chyme, and now finally to feces. Feces is mostly water and about 5% bacteria, or the components of that bacterial decomposition. And then primarily made up of all the indigestible and inorganic materials that you have ingested. Of course, one of the classic examples that everybody loves is corn, right? When you eat corn on the cob, don't you see those kernels in your feces afterwards? No, you do not. What you see is the cellulose outer shell of the kernel. The outer shell of a kernel is cellulose, which cannot be broken down by our digestive systems. It's the same type of cellulose that is used to make grass, right, which we aren't able to eat because we don't have the complex digestive systems that a cow or a sheep have, right? A cow has something like 87 stomachs to be able to break down grass. It's constantly spitting up the food and swallowing it down into a different stomach. Well, the outer surface of that corn kernel is that same type of cellulose. So what happens is our digestive system digests all the materials that are on the inside of that, and just that outer shell is that what remains. But that outer shell of the kernel is expelled in our feces. And let's not forget, as we talked about, that our epithelial tissues inside of our digestive system only last on average about 3 days, 2 to 4 days. So those dead and damaged epithelial cells, whether it was from them being damaged by that sharp tortilla chip or that scalding hot coffee that you took a sip of in the oral cavity or anywhere else along the way, they can be part of it as well. All right, so these are the components that form our feces. Absolutely. So you see that all the time in crime shows where if someone finds a fecal sample, they are able to get a DNA extraction from that. And yeah, typically it is the DNA from those epithelial cells. Because as we know, it's either going to be a simple columnar or it's going to be if the defecation process of coming out the rectum, as we'll talk about, it's passing through a non-keratinized stratified squamous epithelial tissues. So cells can be shed that way as well. So absolutely. So it's going to be those epithelial cells that are used for the DNA sample. All right. And then, like I said, the pigment. Pigment can, of course, be, yes, then the homeless guy comes and stuffs it. Pigment is primarily stercobilin, but also food can cause pigmentation to it as well. Certain foods, if you've ever gone to a birthday party where the cupcakes or the cake has had a black icing on it, often that can cause a green pigmentation to the feces of your kid and stuff like that afterwards. There was this great story about, gosh, 25, 30 years ago about some kids who were there were some kids who were having bright pink, I haven't heard about the hot Cheeto poops, but bright pink feces. And there was all these super expensive tests and things like that were done. And while the kid was monitored in the hospital, it went away. And then as soon as they went home, it came back again. And it turned out they were eating those blueberry cereal, that blueberry cereal that has those pink marshmallows in it. The original pigment that they used in those was a pigment that wouldn't be broken down by the digestive system. So the kid was eating like 3 bowls of those every day, and the pigment was building up and ended up coloring their feces. So yeah, all sorts of things can cause coloration to our feces as well. So exactly that first black poop that comes out of the baby is more of a flushing of the material that is in the large intestine from being in utero. So that's why once the real food starts coming in, then you don't get that sticky tar-like feces from that the same way. It's more of a flushing and starting up of the digestive system from that with the babies. Yeah. All righty, and that brings us to the expulsion of that fecal matter. That expulsion takes place here in the specialized distal region of the large intestine known as the rectum and the anal canal. Again, we have this tube like we expect in the rest of the alimentary canal, but obviously there are going to be some big differences or some specializations. One of them, as we also already talked about, is in the epithelium. Wow, I really cannot talk and type today. I'm usually pretty bad at it, but today it seems to be really bad. Shouldn't have had all that Jägermeister for breakfast. All right, here in the rectum, at the distal end of the rectum and the anus, what is the epithelial tissue that is going to be formed here, that we're going to start to see here? All right, it's simple columnar for most of the way through. But at the end, in fact, if you look closely, you will see a structure known as the pectinate line. That pectinate line is actually where you see that transition of the epithelial tissue exactly into a non-keratinized stratified squamous epithelial tissue. Excellent. So we have that anatomy there with that pectinate line where that occurs. Notice also near the pectinate line, we get a pouching of the rectum itself, or a condensing of the rectum itself. Or notice as it does that, there are these extensions, these folds at the distal end of the rectum where it comes to the anus. And those are exactly the anal columns. This is for helping to condense and express that fecal matter out of the anal canal in that fashion. Notice also we have these structures like rugae, like plicae circularis, where notice they are made of both the mucosa and the submucosa that forms them, known as rectal valves. Now, we know they're similar to rugae in the stomach, and those rugae allow for the expansion of size. We know they're similar to the plicae circularis in the small intestine, which slow the chyme down and increase surface area. So they must be serving some function. Any idea what the function of these rectal valves are?
Slowing down poop coming out.
Say again?
Slowing down feces coming out.
Partially, absolutely. What these rectal valves, and there are typically several of them here in the rectum, do is exactly, it helps in the segmentation, and more specifically, it helps in the separation of the feces from the flatus, allowing you to be able to release the flatus without releasing the feces. Someone want to explain to me what that means in more common terms? There you go. You don't shart. There you go. Allows you to fart without pooping yourself, or my favorite word, shart. Exactly. It helps to help you so that you do not shart. Exactly. So there you go. Or we can say it, more fancy like, in that it helps us to release the flatus without releasing the feces. Excellent. So those are what those rectal valves do. Perfect. There are a couple other specializations in this region that we can see as well. We'll get to those in a second. Oops, wrong direction. Excellent. Notice, and I'll be honest, I'm not 100% certain. It's always best to use appropriate anatomical terms on the exam, but I'll tell you what, since we're online, if I ask you what the function of the rectal valves are and you can say avoiding sharting, then yeah, I will accept that. All right. So another specialization in this area, and I will be honest, I'm not 100% certain why these are present, but you will notice that that green doesn't show up. There are these very large superficial venous sinuses that are located in the submucosa of our rectum. These super large venous structures are what are known as the hemorrhoidal veins. I'm not sure that if it would cause bowel incontinence, because as we'll see, the release of feces is something, but yeah, my guess is that it would affect your ability to be able to pass gas reliably. So I would say that I'm not sure it would affect your ability to defecate, but it definitely would affect your ability to be able to not defecate while trying to pass gas. And we pass gas actually surprising upon it. So you're actually right. That might cause a form of incontinence. I don't know if any of you bothered to read that book Gulp, but actually in Gulp, they actually have done studies to look at how often and how frequently people pass gas. She has this great story where they talk about these special pants that you wear that have this special monitor for how much gas is released by it and everything. So we actually pass gas a lot more frequently than you would think. So if the rectal valves were damaged, then yeah, it might be possible where we might have a type of incontinence because of that. Yes, it is bad. You should always release it. Kill a baby. Okay, I'm not quite sure it goes that far, but there you go. Exactly. Better out than in. Exactly. All righty. Suddenly it's become middle school all over again. All right, excellent. We're all getting punchy. Okay, so again, there are these hemorrhoidal veins. I am uncertain as to what the full function of them is. However, why we are aware of them is if you have an issue with the consistency of your feces. If the feces is too firm, too much water is being absorbed, that can cause irritation to these hemorrhoidal veins, causing them to become inflamed, causing them to become painful, which of course is that condition we call hemorrhoids. right? And of course, then you get that special circle pillow that you get to sit on, where the goal is to try to take some of the pressure off of the rectum in that fashion, so to ease the comfort of that. All right. Now, the other two things that I want to show you on here are, once again, as we've seen, there are valves that are going to control the movement of substance out of the organ. And just like we would expect to see, one of those is an enlargement. Give me a color that I like. We'll go with the bright red, see if that works. Notice here, we see a big thickened enlargement of the circular muscle layer. This nice, big, thick enlargement of the circular muscle layer is what we call the internal anal sphincter. As it is an enlargement of the circular layer of the alimentary canal, it is, of course, smooth muscle. And of course, if it is smooth muscle, we have an involuntary control for this particular, and actually, I don't like that. Let's do that, and let's do that. Smooth muscle that we have that involuntary control of. And again, let's emphasize this. This is an enlargement of the circular layer of the mucosa, the muscularis externa. All right, it's smooth muscle, it's involuntary. I'll put that up there. And, whoops, grab my red. Oh, that was weird. I want that there. I want that there to emphasize that that's what we're talking about. All right, excellent. But I want you to notice something else. There is a second enlargement of muscle here. This second enlargement of muscle are these muscles located over here. These muscles located over here. And again, our artist has not done the best job of showing us this, but they are actually an extension of our urogenital diaphragm. If you remember back from 430, we have the diaphragm, which of course separates our thoracic cavity from our abdominal pelvic cavity. But we also learned about the urogenital diaphragm. The urogenital diaphragm helps to support the external genitalia and the viscera that is located inside of the pelvis. Right? It helps to elevate and support those structures. It plays a role, as we talked about, in urination and defecation. And does anybody remember what the primary muscle in our urogenital diaphragm is? There we go. Excellent. The primary muscle of the urogenital diaphragm is the levator ani. If you remember back to 430, we talked, for instance, about some of the exercises you can do to help to strengthen this. This helps to support the vaginal canal for females, for instance, upon pregnancy, so that, again, it can provide that structure and support. And what do we call those? Kegel exercises. Absolutely, those Kegel exercises. So here, what we have is an enlargement of that levator ani muscle, an enlargement of that urogenital diaphragm. That levator ani, that urogenital diaphragm. And we call this the external anal sphincter. So we have this external anal sphincter, which we have colored blue. That is an enlargement of the urogenital diaphragm, an enlargement of the levator ani. And because it is a part of the levator ani muscle, what type of muscle tissue is it? Skeletal muscle. And because it is skeletal muscle, what type of control do we have over it? voluntary control. So notice we have not one, but two valves that are going to help us to control the release of fecal matter from our alimentary canal. Great question. No, the levator ani provides that support so that as the vagina is stretched so that the baby can come out, that weakens the urogenital diaphragm. So it's not uncommon for a female shortly after pregnancy, if she laughs or if she coughs or if she sneezes, some urine can be expressed out of that because the force in the abdominal pelvic cavity increases the pressure on the bladder, and from the cough or from the laugh, and the weakening of that urogenital diaphragm allows the urine to escape. So things like that can occur. But in extreme conditions, the actual vaginal canal can actually prolapse, where it will actually descend out of the urogenital diaphragm and descend down, and then has to basically be surgically put back into place. And so by strengthening those levator ani muscles, by doing those Kegel exercises, it can help dramatically in the recovery. Right. So basically you just, and again, for those of you who've never had kids before, never had pregnancy, never thought about this before, just think back to Friday night, way back in ancient times when we used to go to places like bars. Right there, you've had your 4th Long Island iced tea and you really need to urinate. However, the problem is the line's 50 deep to the bathroom and there's some perfectly good bushes outside. So there you are outside, urinating in the bushes. And as you urinate during the bushes, suddenly that cop spotlight hits you. Of course, at that point, you have to stop the flow of urine so that you can run. Right. And that ability to stop, that muscle contraction you're making to stop the urine flow, that would be the contraction of the levator ani muscle. Right. So that's that action that you're taking. And that's basically what a Kegel is. You contract that muscle, hold it for a count of 10, and then relax it. And then you do it in sets of 10. Right. And you can do it all the time. You can do it, when you're pregnant. You can do it while you're sitting in a red light, while you're standing in line at Starbucks. I've done 4 or 5 of them while I've been sitting here right now. Again, you can do that to strengthen that. No, it wouldn't weaken during the period because the expulsion of the menses isn't going to stretch the muscle and weaken the muscle in the same way that passing a baby would weaken the muscle. So typically, the levator ani is not weakened from the menses that occurs. Well, but see, if you're not actually making those contractions, they don't actually know you're doing it. So whether you're making eye contact with them or not, right? It's not like when you're at the gym doing those muscles for the adductors, right? When you're on that torture machine, that one's the one you don't make, eye movement with. But if you're doing Kegel exercises, there's no way for anybody to know from the outside. No, because again, the levator ani elevates it, so it may play a minor role in the expulsion, but primarily it is contractions of the uterus and also the abdominal muscle producing the pressure to force the baby out. I guess as the baby's expulsed, if you retracted the levator ani, it might lead to some defecation being, some feces being released during that. But so yeah, I guess it would do that. But that, again, that's the goal isn't to retract that. It's more of I would think it's more of the intense pressure that you're building up inside of the abdominal pelvic cavity that's going to force those things out. Both the baby and the feces. All righty. Excellent. So now we know the pieces that are in place. What we need to do is talk about the process of defecation, how it is that defecation occurs. Now that we know the anatomy, we can talk, and we did all that, we can talk about the process. Here's the pretty picture, but let's go through it in the drawing by drawing it ourselves first. All righty. So here we have our sigmoid colon that feeds into the rectum. And then, like I said, there are 2 sets of muscles. There are the internal anal sphincter, which we will do in green. and the external anal sphincter that we will do in red. And they kind of overlap each other. The internal ones are inside, the external ones are outside, as the name kind of indicates. And again, I want to emphasize that the, oops, too big. Internal anal sphincter is smooth muscle that with involuntary control and the external anal sphincter is skeletal muscle and involuntary control. Excellent. All right, so as we've talked about, there are going to be mass movements, there are going to be peristalsis, there's going to be the releasing of the valves, and what's going to happen is feces is going to, no, let's stick with blue since we started with blue. Feces is going to be entering into the rectum. As that feces enters the rectum, it causes the rectum to expand. And as the rectum expands, it stimulates stretch receptors. Those stretch receptors are going to trigger both a short and a long reflex. So the stretch receptors trigger both a short, or remember what we call intrinsic, and a long, or what we call extrinsic reflex, or really both. Reflex is plural. So it is going to trigger both a short and a long reflex, a local, right, or intrinsic versus a long versus extrinsic, which is going to involve the central nervous system. Let's talk about the short reflex first. The short reflex, basically the signal of that stretch, basically triggers increased motility of the sigmoid colon. So here we go. One, triggers increased motility of the sigmoid colon. That, of course, is going to bring more feces to the rectum, causing it to stretch more, causing it to send an even bigger signal, and things along those lines. But at this, it is also going to cause contractions of the rectum, and it causes the relaxation of the internal anal sphincter. So we're bringing even more feces to the rectum. We're getting the rectum to start to contract, and we get that relaxation of the internal anal sphincter. We sense these things. This is that sensation that we need to defecate. Now, of course, when you feel that sensation, do you instantly defecate right away? Or do you defecate even before you're aware of those sensations? No. And the reason for that, this is that you feel the sensations. You feel that contraction of the rectum. You feel the expansion of the rectum. You feel the relaxation of the anal sphincter. You feel the need to void. But you don't void instantaneously because of the long reflex. Remember, the long reflex is going to involve the central nervous system. In particular, this is a parasympathetic pathway to the sacral that's not sacral. To the sacral spinal cord. So this is a parasympathetic reflex that takes place in the sacral region of the spinal cord. And basically what this parasympathetic reflex does is, not surprisingly, two things. It is going to increase the motility of the colon, again, to move more feces towards the rectum for release. But the second thing that this long reflex does is it constricts the external anal sphincter. So that while more feces is coming to the rectum and we feel the need to void, that constriction of the external anal sphincter stops the feces from coming out. Now, we are born with this reflex. However, as an infant, is our external anal sphincter necessarily strong enough to be able to hold back the forces of that? No. It takes time to strengthen and gain that control of that external anal sphincter. So what happens for the first year and change of your life? Well, yeah, you get to wear diapers. Hopefully not too much of an explosion, but you get to wear diapers because you can't control the release of feces. When that rectum contracts, when that internal sphincter opens, the feces comes out. Of course, the other side of it is at the flip side. Grandpa, who's gotten so old and hasn't been doing his Kegel exercises, has had the weakening of his levator ani muscle. And so now while he has the cognitive control, the neural pathways are in place. Does grandpa necessarily have the strength in his levator ani, in his external anal sphincter anymore to be able to stop the loss? Nope. So you start your life in diapers and you finish your life in diapers, right? Yep, got to work it out. Feel the burn. There you go. So you're feeling the need to void, but you're not voiding as a result of it. So of course, what happens is you get yourself to a socially appropriate location. Once you get to that socially appropriate location, then you consciously relax the external anal sphincter. And when you consciously relax, that external anal sphincter and both sphincters are open, then defecation occurs. And that can be part of the problem, because it's great to feel that need to void and then be able to quickly get to a bathroom and void. But what happens when you feel that need to void your feces and you realize that you just drove past the sign that says it's 35 miles to the next rest stop? All right, you can either pull over to the side and find a bush, or you can drive 35 miles. And you drive the 35 miles and you finally get down to, get to the rest stop, and you drop trow at that point. And then at that point, sometimes defecation can be difficult. The reason it's become difficult is that in time, it's not so much that the signal is blocked, but you absolutely have the right idea. The problem with this is in time, we habituate to the signal. So again, we have this sensation that we need to void. We have this stretch of the rectum and that is causing the relaxation of the internal anal sphincter. But when we habituate to that signal, then what happens is the internal anal sphincter closes again. And if it closes, are we able to voluntarily open it up so that we can get the feces out? Not necessarily, right? So that can be more challenging at that point. We may have to struggle a little bit more to void, have to do some other things to help to assist in that. Yes, Ryan, you had a question. So Kegels might help grandpa or someone who had a weakened levator ani if the reason for the incontinence is indeed a weakness of the external anal sphincter, then potentially doing something like Kegel exercises could help in that situation. Part of it is also, though, can be a loss of the cognitive control as well, in which case they would have trouble doing the Kegels or being able to even make the Kegel exercise type of motion and be able to do it enough to help to so in some instances, the short answer is in some instances yes, but it isn't necessarily going to be a cure-all, right? We're not going to have a bunch of Kegel gyms where old people can get rid of their diapers and stop having to depend on Depends because of that. Yep, sorry. All righty. So the problem is, in time, we can habituate to that signal. However, does that mean it goes away forever and we never have to defecate again? No, of course not, because what's going to happen is that we will continue to fill the rectum. And as we continue to fill the rectum, it stretches more and more. And what happens is it needs a next critical point of stretch. And now the contractions become more powerful. But now you're only 20 minutes from the rest stop, so you still have to hold it. And eventually it habituates and goes away. But in a few minutes, as the rectum continues to fill, it becomes more and more powerful. And is it possible that these contractions can become powerful enough, that the pressure inside the rectum can become powerful enough where it can overcome the voluntary contraction of your external anal sphincter and force the feces out, whether you want it to or not? Yeah, absolutely. If the pressure inside of the rectum gets too high, then that can actually cause, well, at that point, I don't think it's a shart anymore. I think at that point, it's just a full on the other one. So a defecation. So at that point, you're defecating whether you want to or not. All right. Exactly. I like that log. Excellent. Now, again, the goal is for this defecation to occur efficiently and easily. However, there are things that can be done to assist this. So there are things that we can do to make this process easier to occur. Let's change colors one more time. Let's use purple this time. For example, as we talked about, elevating the levator ani, that urogenital diaphragm can help in the expulsion. By elevating, it helps in the expulsion of the feces that way. Another common way that we can help in the defecation process is what is called, well, let's do this first one first, contraction of the abdominal muscles. Contracting the abdominal muscles increases the pressure inside the abdominal pelvic cavity, and that increase in pressure can help to force the feces out. That's kind of what we were talking about with the childbirthing process. But the other maneuver that is used in birthing and also used in defecation, the position can influence and affect it, because again, but as you sit or squat, it actually increases the pressure, not so much gravity, but you're increasing the pressure in the abdominal pelvic cavity to help to force the feces out. So I'm not sure how much of a role. Gravity, I think, helps to keep it from getting on you when it comes out. I think that's one of the huge advantages of that. But I don't think, there you go. That's the one I was looking for, I was going to mention. And that is the Valsalva maneuver. Oops. Excellent. Valsalva maneuver. What is that, Ryan? You mentioned it. Do you know what it is? Don't worry, I'm not going to ask you to demonstrate it on camera. I just was curious if you knew since you came up with the term.
I mean, I know what it is. I just don't know how to
Describe it.
describe it.
Okay.
Yeah.
Basically, what one of the goals of assisting in defecation is to increase the abdominal pelvic cavity pressure. In elevating the levator ani not only helps to extract the feces, but also makes the abdominal pelvic cavity smaller. You make it smaller, pressure goes up. Contracting the abdominal muscles constricts the abdominal pelvic cavity, volume goes down, pressure goes up. The other thing we can do to help to decrease the size of the abdominal pelvic cavity and increase the pressure is to force our diaphragm down, right? So the goal is to force the diaphragm down. And as you force the diaphragm down, that decreases the size of the abdominal pelvic cavity, increasing the pressure and help it to come up. So how do you force your diaphragm down? You use your lungs. Basically what you do is you fill your air with lungs, or fill your lungs with air, right? But then what you do is you close your glottis, right? What we do is we close our glottis, we close the airway so that that airway can't, so the air can't come out. So that then when we try to force the air out with our glottis closed, instead of the air coming out, the air pushes down. So you, and that's that kind of bearing down. What you're doing is you're closing that glottis and you're using air to force down the diaphragm, and that increases the pressure. So that's when you get those great defecation faces, right? As you're trying to force that air down with that Valsalva maneuver. And again, it works well with labor as well too. Again, you're basically, it's that bearing down, but basically what you're doing is you're using air and forcing that air down to help to expulse the feces.
So why would you say the Valsalva maneuver is important for weightlifting and stuff?
So with heavy weight lifting, what you are looking for is the problem. So one of the concerns that you have when you're doing excessive weight lifting, when you're lifting large amounts of weight, is again, what happens is you are increasing the pressure in the abdominal pelvic cavity. And as we talked about, by increasing the pressure in the abdominal pelvic cavity, that can cause straining on the muscles and actually can produce a hernia. So what that same kind of bearing down does is it helps to basically condense the muscles and get exactly engaging your core, squeezing those muscles, compacting those muscles to help to limit the expansion that takes place. It's similar to like using a weight belt. You use a weight belt to stop the expansion of the abdominal pelvic cavity so that you're not getting that stretch, you're not getting that weakness of the walls, and you're not getting that hernia as a result of it. So it's all about helping to provide that protection in that way. All right. Excellent. So here's the pretty picture that goes along with that defecation, but I think we did a good job of describing it. Like I said, notice this is that positive reflex. That short part in particular, we get that stretch causes more feces to move from the sigmoid colon, which causes more stretch, which a bigger signal. And so we get that positive reflex. And again, there's two parts of it. There is a local portion, and there is that long portion that goes through the sacral spinal cord through our parasympathetic nervous system. Right, so we stimulate contraction of the wall, we relax the internal sphincter, we constrict the external anal sphincter so that defecation doesn't occur right away. Instead, we wait till we get to a socially appropriate location, and when both sphincters are relaxed, voiding can occur. If we don't relax that external sphincter, habituation will occur, but the feces is still there. And we're going to get an increase in pressure and an increase in the contraction, and the pressure becomes greater and greater. And remember, as we said, if the pressure gets high enough, like 55 millimeters of mercury, then it can actually force the external sphincter open, forcing defecation to occur, whether you want it to occur at that point in time or not. And then we also talked about ways to assist in that defecation process as well. And with that, we are out of the alimentary canal. Holding it causes an increase in pressure, yes. And the greater the pressure, the more force it's going to force it open. Absolutely. So again, your book's got some great tables that talk about everything that happens in all of the organs of the alimentary canal. And with that, we have taken our mystical, magical journey from cheeseburger to feces and everything in between. All right, questions on that? So impacting of the fecal matter can occur because of several reasons. One can be a decrease in the motility in our large intestine. If the large intestine slows down in its motility, what happens is the feces stays in it for too long and too much water is absorbed, and then the feces becomes hard and the large intestine can become impacted in that way. So that can be one of the problems that can occur. This can be a major issue for individuals who are paraplegics, for instances. With a paraplegic, they have less again, it's still involuntary, but they have less central nervous system control of these. And so they're not able to stimulate it with the parasympathetic the same way. So instead, those intrinsic, because there's still that intrinsic reflexes, are much slower. And so it can cause a problem that way. Drugs. So again, probably Elvis. Again, whether he actually died of an impacted colon or not is I'm not sure if that if I'm not sure if that's was the cause or stated as the cause of death, but he definitely had a very large impacted colon, and his was primarily caused by narcotics. Taking certain narcotics can decrease the motility of the large intestine, and that can cause impacting, and that can cause problems with defecation. So a lot of times when you go in for surgery, and it's a general surgery where you are anesthetized, that a lot of the anesthetics they use can decrease motility. So one of the things you have to do before you're allowed to leave the hospital is defecate. So those are some of the things that can influence it, impact it as well. Yep. Any other questions on this particular subject? All right, so here's the game plan. It looks to me that it's about 2:45. Let's just take a quick break, a 10-minute break. That will give a chance for anybody who wants to run screaming from the building the chance to do that. But let's come back at 2:55, and at 2:55, we can switch gears into our Q and a review. And I will start the recording at that point, so that we make sure that we have it. And I will happily answer any questions about anything lab, lecture, exam, or any other material that you need some help with, so that we can hopefully have everybody be as successful as they want to be on that exam. Yes, definitely some pain medications can cause constipation as well. Absolutely. And primarily through decreasing the motility of the large intestine. Yep.
I was always curious how that works.
Yeah, that's primarily how it does it. Yep. All righty. So excellent. Let's take a quick 10-minute break, stretch, go to the bathroom, get something to eat, get something to drink, stretch, and we'll meet back here in 10 minutes. And then, like I said, I will answer any questions you guys have for the exam. And remember, have questions. A review is not me telling you what I think is important. That's what I do every day in class. And remember, if you don't ask questions, then I assume everybody's mastered the material and I make the tests harder. All right, I'll see you back here in 10 minutes. All righty. So the time that is left in the class is yours. This is your opportunity to ask any questions that you have to help you to prepare for Tuesday's exam. So what can I do to help you to master this material and get the grade you want on the next exam?
I have a question.
Sure.
So about rugae in the stomach,
Mhm.
you said they comprised of mucosa and submucosa, or?
Yes, mucosa, they're folds of the mucosa and submucosa. That is correct.
So that's how we distinguish from the Actually, no. Okay.
Yeah, so gastric glands are extensions within the mucosa only. And remember, the easiest way to be able to distinguish that is going to be in terms of the muscularis mucosa. So remember, the muscularis mucosa is going to be that little bit of smooth muscle that is going to line the mucosa. So we have the mucosa, which is all of these huge, thick with all the gastric glands and gastric pits that are coming down in through it. So we have the surface of the mucosa here, and then all the gastric pits that come down into it. However, our submucosa normally just chills and goes along, but there are going to be portions where it extends out. And so what's going to happen is that's going to form this big, huge fold that sticks out of our stomach's wall. And in that stomach wall, that's going to be that ruga.
Okay.
So basically find the muscularis mucosa, and that'll help you to distinguish when you've got that ruga that extends out.
And one more question. You didn't mention the pyloric antrum.
So so remember we talked about that mostly when we were talking about the histology. The antrum is basically just the funnel portion of the pyloric region that feeds into the sphincter, that big, huge sphincter. And so remember, when we looked at it histologically, the wall of the pyloric antrum is basically almost exclusively containing just mucus glands instead of the entire normal gastric pit that has, the parietal cells and the chief cells and the enteroendocrine cells. So that was how we were able to easily tell the difference between the pyloric region and, for instance, the fundal region or the body. Is it possible to tell the difference between the fundus and the body? Yes, but I think those differences are a little bit more subtle. So I'm not worried about those distinctions, but being able to distinguish between the body and the pyloric region is something that definitely you need to be able to do histologically.
So when you're asking for the region, I can say just pylorus, or I should say pyloric antrum.
Pyloric region would be fine. Yep.
Okay.
I certainly wouldn't mark it wrong if you put antrum, but again, antrum is just entryway. So it's the funnel-shaped entryway into the small intestine.
One more question.
Certainly.
Swallowing reflex is triggered by uvula touching the wall of the
No,
cavity, or bolus who pushes the uvula. How?
Neither. The swallow reflex or deglutition reflex is caused by the bolus coming in contact with the arches, not with the uvula, but with the arches that line the oral cavity. So the palatoglossal and palatopharyngeal arches. So it's actually the contact with the arches and not the contact with the uvula that triggers the reflex.
Okay, yeah, now it makes sense.
Yeah.
And last questions for now.
Okay.
What is the right order of 6 basic digestive processes?
See, remember, that's the key. There isn't a correct order. Those 6 functions aren't in order. Now, there are certainly some things that have to happen first. Obviously, you have to ingest food before you can do anything else to it. Obviously, once you're done with everything, then you're going to defecate the food. So yes, one of those has to come first, one of those has to come second. But all the other things basically occur in all sorts of different locations, right? In the mouth, you have absorption, you have chemical digestion, you have mechanical digestion, you have propulsion. So all of those types of functions that are occurring aren't in order. We don't do all of the propulsion first, and then all of the chemical breakdown, and then all of the mechanical breakdown, and then all of the absorption. All the rest of those functions happen at all sorts of different locations along the way. So there isn't a correct order to those 6 things because they're not occurring sequentially.
Okay, thank you.
Yep, great question. How about some more?
Can can you go over the histology for the salivary glands one more time?
Sure, absolutely. I think we have that in the lecture, so let me go back and find it more easily rather than just scrolling through all the slides. All right, I'm going to, well, we'll do it this way, this will work. So when you are looking at the salivary glands, really, when you're looking at most exocrine glands in the body, there's basically two things that you're looking for, because after all, exocrine glands are comprised of two components, ducts and secretory structures. Okay, so that's the first thing we always want to look for. We always want to look for ducts, and we always want to look for secretory structures. Do we see those two things here in this illustration? This is the point where you all say yes, absolutely, excellent, perfect. All right, which of those two things that we talked about would this here be? Excellent, that's a duct. All right, and remember, all ducts do is carry the secretion out of the body. They're the PVC pipe that moves it. They're the straw that it passes through. It doesn't change it, it doesn't make it, it doesn't mattify it or anything. It's just the passageway out. Basically, pretty much everything else in this field of view is a secretory structure. Now, I say pretty much. This down here is actually a blood vessel. Here's an adipocyte. Here's an adipocyte. Here, these are adipocytes. So again, not truly everything, but basically everything else we see in here is a secretory structure. And of course, the secretory structures make the secretion. So secretory structures make the secretion, ducts carry the secretion out. Now, with salivary glands collectively make saliva. And as we talked about, saliva basically has two components to it. What are the two components to saliva?
Mucin and serous fluid.
There you go, serous fluid and mucin, absolutely. So those glands that are going to be associated with making saliva are going to have secretory structures involved in making one or the other of those types of substances. Now, as I look here at this particular gland, do all of my secretory structures look the same? No. Notice some of my secretory structures are very light. The cells are very light in color, and they're very irregular in their shape. Other secretory structures are much darker in color, much more compact, and much more uniform in their shape. So let's start easy. Color. Color is usually not something we pay attention to on histology because it's usually an artifact and usually not important, except when it is. There you go. And Ashley's hit it on the head. When we have cells like this, let's not use that color. When we have cells like this that have this very light coloration to it, that is an indication that these are the cells that are making mucin. Conversely, these cells over here aren't making mucin. And so if they're not making mucin, what must they be making? Serous fluid, right, absolutely. So the mucin is light, the serous fluid ones are dark. So here we happen to have two different types of secretory structures making two different types of secretion. One is making mucin, one is making serous fluid. Now, as I also think back to 430, not only do I remember that exocrine glands are made up of ducts and secretory structures, but I also remember that there are two possible shapes for my secretory structures. What are the two possible shapes for my secretory structures?
Balls or tubules.
All right, ball shape or long, narrow, thin shape. And the ones that are long, narrow, and thin, we call tubules. The ones that are ball shape, we call alveoli or acini, right? We can use those terms interchangeably. Excellent. So based on what we see here, since I have two different types of secretory structures and they're therefore going to have two different types of shapes, which one look more like they are uniform ball shape and which one looks like they may be tubes cut at any sort of different angles? Remember, the trick with a tube is if you cut a tube in a cross section, it looks circular. But you can also cut a tube longitudinally. You can cut it at an angle, right? They can be bent. So the tube ones tend to be more irregular in their shape. Whereas when you have a ball-shaped structure, no matter how you cut it, you pretty much have a ball. So as we look at these things, are the things making serous fluid the acini, or the things making the mucin the acini? There you go, exactly. The things making the serous fluid tend to look more ball-shaped, and indeed, these are acini. These are the acini producing serous fluid. Whereas the irregular shape, light-colored secretory structures are the tubules, and they are producing mucin. So notice this particular salivary gland has mucus-producing tubules, and it also has serous producing acini. So that means the composition of the saliva that this is going to make is going to be even parts mucus and serous fluid. And which of our three salivary glands produces equal amounts of mucus and serous fluid?
Submandibular.
Submandibular. So this gland right here is the submandibular gland. Now, two more pieces of information we need to know. For all of our glands, calling it a mucus tubule would be fine, or a tubule, secretory tubule that makes mucin would be fine. Calling it a serous or acini that makes serous fluid is fine, or a serous acini, absolutely. Those are absolutely appropriate terms you could use for them. Now, the last thing, for any gland, we need to also know its structural classification and functional classification. Let's start easy because the functional classification of all of our salivary glands are the same. What is the functional classification of this gland?
Merocrine.
Merocrine, which basically means it uses the exocytosis to release its contents. So the second question is, what is the structural classification of this one?
Compound tubulo-alveolar.
There you go, a compound tubulo-alveolar gland. Excellent. Now, normally when we think compound, you have to see two or more ducts and two or more secretory structures. We definitely see two or more secretory structures, but we only see one duct. Now, notice this is a very high magnification view, so it's not a stretch to think that there might be another duct somewhere in this gland. But more importantly than this has both tubes and alveoli. And the only type of gland that has both tubes and alveoli in the same gland is a compound tubulo-alveolar gland. So even if I didn't see any ducts, the fact that I saw two different types of secretory structures that make two different types of substances is a dead giveaway that this is a compound tubulo-alveolar gland, which is our submandibular one. Now notice, if instead we look at this gland, again, we are looking for ducts. We are looking for secretory structures. Do we see ducts? Yeah, absolutely. All right. Here's one. Here's one. And really, at that point, do I have to keep counting? No. Once I found two or more ducts, I know this must be a compound gland. That's going to help me. And now I look at the secretory structures. For the most part, as far as the eye can see, do I see one type or two types of secretory structures? Pretty much one. Which of the two types of secretory structures am I seeing? Mucin. I'm seeing cells filled with mucin, and they're irregular in their shape. So you guys combined are absolutely correct. We have a bunch of mucus tubules. So the saliva that this particular gland produces is going to be almost exclusively filled with mucus. Of course, it uses that merocrine mode of secretion. Since it's just mucin tubules, this, of course, structurally is a compound tubular gland. And which of our three salivary glands is primarily just making mucin? There you go, sublingual. Do we see ducts? Yep. Do we see secretory structures? Yep. Again, we see more than two ducts. We know this one's compound. But what do we notice about the secretory structures here?
Alveolar.
All alveolar shape, absolutely. And not only are they more circular and ball in shape, but what else do we notice about them? Yeah, they're dark in color. And since they're dark in color, we know they're not making mucin. Instead, like Ashley said, they're making serous fluid. Absolutely. So here we have serous acini, many serous acini, and many ducts. So a compound acinar or alveolar gland. And what's the composition of this saliva going to be like? Is this an equal mix of mucin and serous fluid?
No.
No, it's going to be just mostly watery serous fluid, absolutely. So which of our three salivary glands is this? Parotid, excellent. And I don't know how many of you have been watching the news over the past 2 days, but I don't know if you saw or heard, but researchers have found what they believe to be a fourth paired salivary gland. They believe they have found an additional salivary gland that actually produces saliva that is released into the nasopharynx. They believe it is draped over the auditory tubes, which is why it hasn't been found up to this period of time. They were doing some special testing with some very high resolution MRIs, looking at cancer cells and things on those lines, and they just found what they believe may be a fourth set of salivary glands. So should you have to take this class again in a couple of years, because it'll probably take that long before they've had a chance to name it and learn more about it, you may be learning about 4 sets of salivary glands. It's really cool. I know they try. I know was like a year ago. It's like the one good thing that came of COVID. Like a year ago, they were trying to say that there was an additional organ system. They were trying to say that all of the connective tissues that hold the body together form some special organ or organ system called the interstitium. And again, it's just, it's, in my opinion, that's just anatomists trying to sell more books, right? You're overcomplicating things more than it needs to be. Yeah, does the connective tissue work together? Are they interconnected across organ systems? Sure. Can that affect how we treat certain conditions? Probably should. But again, it isn't a new novel piece of information. This is the first new novel piece of information that has been found in a while. I think the only thing that I can think of previously to this is a few years ago, they found an additional ligament in the knee that some people had and some people didn't. And so they're finding that that was affecting how people were recovering from knee surgeries as to whether or not that ligament had been damaged and they had that ligament or not. So it's really cool that it might be in there. Not sure about a sharp pain. What I would say is that, for instance, sometimes when you eat something really sour, you kind of feel like a sucking sensation that can occur in your cheeks. That sucking sensation is from the extraction of saliva from your parotid gland. So that is a common experience. A sharp pain, I'm not so sure about that. A sharp pain when taking a bite, I think I would be concerned has more to do with the joint or ligaments or something along those lines. I guess it's possible. Again, not my area of expertise, but that sucking sensation is a common sensation that I'm aware of, not necessarily a sharp pain one. All right, so there you go. Those are the 3 confirmed salivary glands and the histology of that. Any questions on those? All right, what's next? Okay. So again, you go to your textbook, you go to your histology atlas, you go to your lab manual, and then after you're comfortable with this, you go to the almighty Google. And when you do that, you will find some pictures that look something like this. I like this picture because it is a relatively low magnification that really does a nice job of showing the compartmentalization of the liver. Remember, the liver is made up of these structural and functional units that are the liver lobules, these hexagonal shape structures that have the central central artery, the central vein at the center of them, and then have all of these hepatocytes that are radiating towards them, towards the middle. And all the spaces in between is where we have those sinusoid capillaries. And then separating each individual liver lobule is these connective tissue septums. And within these connective tissue septums is where you're going to find those structures involved with feeding blood into the liver lobule and also receiving the bile from moving out. So this was that because I like the low magnification view of it. Here, what they've done is they've dissolved away the blood vessels so that you can see a lot more just of those hepatocytes. Notice they call them here the cords of liver cells. You don't have to worry about that term cords, but those are the hepatocytes that are forming those rows. And in between the rows, you can see the big open space where those sinusoid capillaries would be located. Remember, also in this space over here out in the connective tissue septum would be where all of the blood vessels that feed in and the bile duct that receives the bile of those hepatocytes coming out would be located. And if we were to take an up close view at a region like that, we would see something like this. And this one, like I said, is probably about as perfect as I've ever seen when I did the search for this just the other day. And it does a great job of showing those 3 structures, the portal triad structures that would be there. And as we remember, those 3 structures, what is structure 1 here on this list?
Vein.
Vein, although let's be more specific. It is a branch of which vein? Number 1 is a branch of the...
Is that the hepatic portal vein?
Hepatic portal vein. Excellent. What is the condition of the blood in this hepatic portal vein?
Oxygen poor, nutrient rich.
Excellent. Perfect. Oxygen poor and nutrient rich. Spectacular. Excellent. And that feeds it into the sinusoid capillary so that the hepatocytes can process it, make it nutrient appropriate. All right, which brings us to vessel 2. What is vessel 2 of our triad here?
The hepatic artery.
Yeah, it's a branch of the hepatic artery. And what is it bringing to the party that we need?
Oxygen rich, nutrient adequate. or appropriate?
True. Yeah. And again, yes, it is going to have nutrients in it because it is artery, but really the important thing that it's bringing to the party here is that oxygen. Processing of the hepatocytes requires a tremendous amount of energy. So really the key thing that the hepatic portal vein brings is the nutrients. The key thing that the hepatic artery brings is the oxygen so that the work can be done. And so these two bloods mix together in the sinusoid capillary, providing the oxygen necessary for the hepatocytes to do their work, and making the nutrient levels appropriate, storing excess stuff and expressing things that are deficient. So that when it finally reaches that central vein at the center of the lobule, it is used up the oxygen, so it is now oxygen poor, and it is nutrient appropriate. It can go back to the heart, and it can go get the oxygen that it needs from the lungs and then be distributed throughout the body. However, making the blood nutrient appropriate, as important as it is, isn't the only function of the hepatocytes. Those hepatocytes are also making bile. They release that bile into tiny little tunnel-like structures. And what are those tiny little tunnel-like structures called again?
Biocaniculi?
Biocaniculi, absolutely. And those canaliculi feed into structure 3. And structure 3, of course, is
Bile duct.
the bile duct, right? So those are the 3 portal triad structures. And the other key, so yes, hepatic triad is made up of hepatic portal vein, hepatic artery, and bile duct. That is correct. Oh, great question. Yes, I guess, well, no, it wouldn't be a venule so much. I think if you really wanted to be technical, I would say a branch of the hepatic portal vein. Technically, I think it would be a hepatic arteriole, but you could just say a branch of the hepatic artery and then the bile duct. So I think that would be sufficient. I guess technically it would be an arteriole. I'm not sure we would call it a venule because remember, venules are what receive from the capillary and this feeds into the capillary, but this isn't a vein, but this is a vein. So I'm not sure actually how they would name that. But again, you're not going to see them written that way. Typically, it's considered a branch of the hepatic portal vein and a branch of the hepatic artery. No, you don't have to put you don't even have to say branch in the name. I would be fine with that. Yeah, it's just indicating that they're rows. The same way they use that term cord plates means the same thing. It's just a row of those cells. Absolutely. Now, the one last thing I want to say about our portal triad is how were we able to distinguish the 3 of them?
Their size and the muscular layer around the,
Excellent. So yeah, so the venule typically, the vein, see now you got me doing it. The vein is typically going to be largest, the lumen. The artery is going to have a thicker muscular wall. But notice both of them are lined with simple squamous epithelial tissues, whereas the epithelial tissue that forms the duct is going to be a simple cuboidal epithelial tissue. So the epithelial tissue here is much more prominent, much more obvious. The muscle layer, much more obvious. The size, typically very obvious. So each one of them has something very obvious about them that should help you to be able to distinguish them. All right. Questions on that?
and what's the type of muscle layer around the
Well, again, remember it's an artery, so that would be the tunica media and a circular layer of smooth muscle. That is correct. All right. Any other questions on this? All right. What's next? Flow of what? Oh, So again, one of the points that we talked about, and I want to make sure that we emphasize, you're right, is the directionality of the materials. So as we've talked about, from the artery and the vein, the blood enters into the sinusoid capillaries from the artery, from the vein, and that blood travels through the sinusoid capillaries towards the central vein where it is processed. So again, you have oxygen-rich blood coming from, oops, that's not enough. Oxygen-rich blood coming from the artery. You have the nutrients coming from the vein. And again, it's nutrient-rich coming in there, mixed together. The hepatocytes process it. So by the time it reaches the center, it is nutrient appropriate. And again, notice this is happening from all the corners of this. So let's do that. From all the corners of this lobule. So all of the blood is coming towards the center through those sinusoid capillaries towards the middle as it is being processed. Ironically, I used green for that because it would have been better for the bile, but that's all right. At the same time, our hepatocytes are making bile, and as they're making bile, they're sending it out to the outer wall of the lobule where the connective tissue is and where that bile duct is located, so that bile duct can collect that bile as it is moving out of the lobule. So you have bile moving in one direction and blood moving in the other, but remember, they're not flowing through the same channels. The blood is traveling through the sinusoid capillaries and the bile is flowing through the bile canaliculi. So it's not like the two streams are crossing, they're passing in different structures and going in different directions. Did that answer your question? Yep, excellent. All right. Other questions on the liver? All right. What's next? I got my little Batman stress doll. What's next? Is that it?
Can you explain the absorption of the carbohydrates, like the glucose going in the cell?
Sure. So you just want the absorptive pathway, or do you want all of the material? Do you just want how it crosses
So,
the apical and basal, or do you want all of it?
yeah, that.
All right. So again, remember we did this at the end of class yesterday. So we did proteins and we did lipids for the first time today, but we did carbohydrates on Tuesday. So that's why we breezed through it so quickly today, but we can go ahead and do it all together. Carbohydrates are broken down into what?
Monosaccharides.
Excellent. Oops. There we go. Saccharides. Excellent. As always, we are going to break them down mechanically and chemically. What is the form of mechanical digestion? Where are the locations? What are the things that we do for mechanical digestion?
Mastication in the mouth.
Excellent. Mouth. Mastication.
The churning in the stomach.
Stomach. Churning.
Segmentation in the small intestine.
Again, that big three that we know is always going to be there. That makes that easy. And with carbohydrates, is there a special type of mechanical digestion that just involves carbohydrates? Yes, no, maybe? No, right? There is a special type of mechanical digestion for proteins. There is a special type of mechanical digestion for lipids, but not for carbohydrates. Carbohydrates is just the first three. Because remember, carbohydrates are pretty easy to break down. Very, very simple, very easy, very basic. There are a few disaccharides that can be absorbed, but in most cases, they need to be broken down into the monosaccharides to be absorbed. Yeah, there are a few larger molecules, but the vast majority of the sugars that we're going to absorb are going to be monosaccharides. All right, chemical digestion.
Salivary amylase in the mouth.
And again, I know it's a stupid question, but where does the salivary amylase come from?
In the mouth.
Be more specific. This is occurring in the mouth, but where is the salivary amylase coming from? Salivary glands.
Yeah.
Again, it seems like a very silly question to ask for this one here, that this is occurring in the mouth with the salivary amylase from the salivary glands. But if you think about, for instance, in lipids, the lingual lipase isn't active until the stomach, but it was produced in the mouth with the tongue. So that's the reason why it's better to be thorough with these things. We want to say where it's taking place, what the enzyme is, and where that enzyme came from. Any others?
Oh, the pancreatic amylase in the pancreas.
Pancreatic amylase.
Oh, and then isn't it the duodenum?
Yep, although in this case, you can just say small intestine.
Okay.
So in the small intestine, but you're right. If you said duodenum, I certainly wouldn't mark that wrong, because that is the part that the pancreas is releasing its pancreatic amylase to. And again, saying the pancreatic amylase is from the pancreas seems silly because it seems redundant. But for instance, when we talk about trypsin, does trypsin tell us where it comes from in the name? No. So there we need to say trypsin from the pancreas. So again, even though it seems silly, it seems redundant. We definitely want to say those things. And then, of course, we can't forget in the small intestine our brush border enzymes. Whoops, yikes. Any others? Nope, can't think of any others either. All right, excellent. So we need to pass the apical cell layer. What's the mechanism to cross, or mechanism, or isms, plural, to cross the apical cell layer?
Yeah, co-transporter.
Excellent. Primarily co-transporters. And what else?
Facilitated diffusion.
Yep. Excellent. So that co-transporter again uses sodium. The facilitated diffusion uses carriers, as opposed to, that's not how you spell carriers. Instead of, for instance, like just channels. Excellent. What is the, I guess for consistency's sake, is there any change to the molecules inside the cell?
No, still monosaccharides.
Right, exactly. So again, right, we say that because remember the lipids changes do take place inside the cell. So if we're trying to be consistent in the way we're learning this, it helps to do that. So nothing happens to it inside the cell. So then we just need a mechanism or isms to cross the basal cell layer.
transporter. It's like a protein, right?
Well, it's another carrier. It's facilitated diffusion, right? Again, on the basal surface, we're not going to bother using ATP to get them out because we've gotten it out of the lumen, which was the big deal. We wanted to make sure that we didn't lose it. It didn't go away in the feces. But once it's in the cell, it's inside the body. And so we don't have to worry about wasting energy to get it out. As it accumulates inside the cell, it'll build up its own gradient. And by facilitated diffusion, it will leave the cell into the interstitial fluid. In the interstitial fluid, what type of capillary does it enter?
Blood capillary.
Blood capillary, excellent. And then, of course, the pathway to the heart.
A strong point. Intestinal vein. Or
Yep, intestinal vein. Those intestinal veins feed into the
mesenteric vein.
There you go, superior mesenteric vein, which feeds into the
I always get lost right here. Is it the hepatic system?
True. So we want to go to the liver. So what is the vein that goes to the liver?
The hepatic portal vein.
Portal vein. Excellent. Remember, this portal vein is basically between two capillaries. That's what the portal systems are, right? Remember, we also talked about a portal system with the pituitary gland. When you have two capillaries kind of lined up, you kind of have this portal system in between. And so the hepatic portal vein takes us to the to the liver. And once we're in the liver, where we now get to process that. And where does that processing take place? Our hepatic portal vein, remember, is part of the triad, so it feeds blood into the
hepatic vein, right?
Before that, remember, we have to process it. And remember, things can only come in and out of a blood vessel in a capillary. So from the hepatic portal vein, we have to go into the special capillary found in the liver. You getting some help in the chat?
Oh yeah, thank you.
Sinusoid capillary, excellent. That sinusoid capillary feeds into what?
The central vein.
Excellent. Now we can leave the liver, and we leave the liver via what? What takes us out of the liver? So the hepatic portal vein brought us, the hepatic portal vein brought us into the liver, and then it is the hepatic vein that takes us out.
Okay.
And that hepatic vein feeds into what? What big blood vessel does the liver wrap around?
The inferior vena cava.
There you go. Which, of course, feeds into what?
Right atrium.
Bingo. There you go. And just that easily, you have answered all the questions, provided all the material necessary to describe the absorption pathway for carbohydrates. Questions on that?
So protein digestion and carb digestion, when it's inside of the cell, they don't change, only lipids change, right?
Correct. All right. Excellent. What's next? Is that it? Our brains are full? All right. Well, there are no more. Oh, Ryan feels full. Excellent. Which seems appropriate for the digestive system. Excellent. Okay, there's a good question. The phases of gastric activity again. I like that. What are the three phases of gastric activity?
Cephalic,
Phallic.
gastric, and intestinal.
Let's see if we can condense information to high points. Cephalic. Where does this phase get its name? Where is the food during the cephalic phase? True, it's in the head, right? But again, remember, that could be in the mouth or mental or something like that. So another decent way to think of this is that this is before the food reaches the stomach, right? So that's our goal. Our goal is of the cephalic is before the food reaches the stomach, right? So that is when this phase occurs. Let's actually do this, see if this works this way. All right, cephalic is before the food reaches the stomach. Excellent. Is it excitatory or, yeah, so it can be sight, smell, taste, thoughts, right? Taste counts as well. Notice taste is inside the alimentary canal, just not in the stomach yet. Absolutely. So all of those are examples of ways that we can start the process. Now, ultimately, for successful digestion, it is nice for it to be excitatory. But does it necessarily have to be? There you go. And there's the key word. It is either excitatory or inhibitory. Can it really be both? I like either. I like that too. It's either or. It's either excitatory or inhibitory. All right. So again, it's going to be one or it's going to be the other. Not only do we need to know if it's excitatory or inhibitory, but we also need to know is, because we're controlling the stomach, is it controlled by the nervous system, the endocrine system, or both? Is it both with the cephalic phase? Yeah, and in this one, it's just nervous only. Or neural, let's say neural. And of course, if it's excitatory, it's going to be the parasympathetic. And if it's inhibitory, it's going to be sympathetic. What is the primary targets of the cephalic phase? Yeah, so fight or flight, rest and digest. So we have that neural control. Assuming it's excitatory, what is the goal? True, it does target the salivary glands, but remember, right now we're talking about gastric. We're regulating gastric control. So when we're regulating gastric control, we really want to focus just on the effects of the stomach. So remember here, our goal is to prime the pump. Get the stomach ready to deal with the food that's on its way. So absolutely, it's going to, again, assuming it's excitatory, if it's excitatory, then it is going to increase the secretions, and it is going to increase motility, the contractions. Absolutely, that is going to be the goal. All right, makes your stomach grumble. Getting it ready. Sure, absolutely, that would be worthwhile to say. If we were talking parasympathetic, knowing that it is the vagus nerve is definitely important. Increasing the churning, increasing the muscle contractions, and is that something that you can sense? Absolutely. Although the secretions can lead to the grumbling sensation as well. And of course, it's a sympathetic nerve. Oops. From the celiac ganglia. If we were trying to talk about where that control is, the pathway is coming from. Increasing the secretions, increasing the motility, getting it ready. All right. How does this relate to step 2, gastric phase? When does the gastric phase take place?
In the stomach, when food gets stomach.
Food reaches the stomach. Is it excitatory or inhibitory?
Excitatory.
Absolutely, it is primarily excitatory. Now remember, as we talked about, in stressful situations, very stressful situations, right, rarely there can be inhibiting. But that's not primarily the goal of it. The goal of it is for it to be excitatory. We need to be aware of the fact that in an incredibly stressful situation, it can be inhibitory, but in here, the goal is primarily to be excitatory. And that excitation of the gastric phase primarily comes from what? Neural control or hormonal control, or both? Both, absolutely. Both neural and hormonal control. For the neural control, where does that neural control primarily come from? Now, is it coming from the vagus nerve, from the parasympathetic nervous system? The sympathetic from the celiac ganglia would be the inhibition of the cephalic phase. So like I said, if you were a vegan and you didn't like it, absolutely. So, okay, be careful. It comes from the enteric plexus. But remember that enteric plexus has 2 components, the submucosal plexus, which controls the glands, and the myenteric plexus, which controls the smooth muscle. And in this case, are we going to want to control just the glands or just the smooth muscle? Or are we going to want to control all of it? Between. The myenteric plexus is between the circular and longitudinal layer, and the submucosal plexus is in the submucosal region of the, in the areolar connective tissue of the submucosa. All right, we're kind of getting into a different essay question by getting bogged down in this enteric plexus and how it works. But the point is, the neural control is going to primarily be a local reflex. So the neural control, let's actually do it this way, is that local reflex using that enteric plexus. All right. But it also can be hormonal as well. What's the hormonal control based on? Where does the hormonal control? What controls the stomach hormonally during the gastric phase? What hormone? gastrin. There we go. Perfect. Gastrin divides control. And our goal now is to process the food. To do that again, we're going to increase the motility and increase the secretions. All right, process that food that is inside of the stomach. And that brings us to our third phase. Our third phase is the intestinal phase. Oh, where'd it go? There we go. In the intestinal phase, when is the intestinal phase taking place? Where is the food during the intestinal phase? Yeah, this is when the food enters the small intestine. Intestinal phase, excitatory, inhibitory. What's the story there? Excellent. And here it really is both excitatory and inhibitory, right? First, oops, there we go. And again, briefly, it is excitatory. But then next, for longer, it is inhibitory. Excellent. For this one, let's start first with the excitatory. Excitatory control, is it neural or hormonal or both? What causes that brief excitation? Anyone remember?
Gastrin.
Exactly. So it's hormonal, right? The small intestine releases intestinal gastrin. And that intestinal gastrin works the same as stomach gastrin. Increases secretions, increases the motility of the stomach. However, the inhibition is the inhibition hormonal, neural, or both?
Neural.
There is a neural.
And hormonal.
There you go. Both neural and hormonal. We can start with the neural first. The neural first basically is that reflex. What was it called again?
Enterogastric.
Excellent. That enterogastric reflex that basically does three things. That enterogastric reflex inhibits the intrinsic reflex. So it inhibits the enteric plexus.
May I ask, when you're talking about enteric plexus, you mean short reflex?
Yes, I'm saying, remember that enteric plexus is that gut brain where it can actually make some basic decisions on what the stomach does. And in this case, it can tell, like we talked about, when the stomach stretches, that signal goes to the enteric plexus. The enteric plexus processes that information and tells the stomach to start to churn. So it's the enteric plexus that actually makes that decision. So what this reflex does is suppresses that. So now the enteric plexus can't tell the small intestine, the stomach, what to do as much anymore. So it becomes less motile, right? It inhibits the parasympathetic, or really basically, if you want to be specific, it inhibits the vagus nerve. So there's less parasympathetic input. That parasympathetic stimulates the digestive function. So this decreases digestive function. And, oops. Oh, I spelled this really bad. And it stimulates the sympathetic pathway, which also inhibits the stomach. So notice all of these things, blah, blah, blah. All of these three things are going to slow the stomach down. Decrease secretions, decrease motility, right? Same thing with the hormones. Remember, there are lots of hormones that can be involved, and they are released based on the components or the composition of the chyme. So if there's a lot of lipids, then we release cholecystokinin. If there's a lot of pH, we release secretin, and so on and so forth. And you should know those hormones and what the triggers for those hormones are and what they do. However, the key, and I'm going to have to cheat and move this one up a little bit so we have room to write. The goal, when we're thinking of goals of this process, the goal is the same. The goal here is to regulate the release of chyme into the small intestine. We want to limit the activity of the stomach. The stomach wants to process all its food and get rid of it. But this is our way of telling the stomach to slow its roll. We only want you to release the material into the small intestine at an appropriate rate so that we can maximize the efficiency of the small intestine. We want to be able to maximize the efficiency of the breaking down of that food, mechanically, chemically, the absorption of it. So we want to control the rate of release from the stomach into the small intestine. Right, and let's make sure we emphasize from the stomach, because after all, that's the point of this. The point of this is we are controlling the stomach. That's what all three of these things are about. 3 different phases for controlling the stomach and what the stomach does.
Does our nervous system remember how often and how much food we intake? Usually, for example, they like, if we often intake.
No, I understand what you're saying. Not really. Like I said, we do have hormones like leptin that are supposed to help to influence our hypothalamus to make us feel sated and full. Although what I will say that, okay, so yes. So one of the things that happens, for instance, as our stomach stretches, the more the stomach stretches, the more powerful of a signal we get to the brain telling us that we are full and feel sated. So yes, you can get neural signals from the stomach, for instance, that can kind of try to trigger the stopping of eating or that sensation of being full so that hopefully you will stop eating. But that's the problem. A lot of times we don't eat because we're hungry, right? So feeling full doesn't necessarily stop us from continuing to eat, which is why, right, on Thanksgiving, we all sit there in pain because we had that third helping of stuffing, even though we didn't feel hungry. It tasted great and we wanted to eat it, and we had to beat our siblings to get to it. So we have to make sure they don't get any. So we had to eat all of it, or whatever reasons they are, right? There's plenty of other motivations for why we eat other than hunger and satiation. So theoretically, those are in place, but as we know, everything is far more complicated than that. All right. Excellent. Good question. Any others? All right. Now are our brains full? Yes. So here's what I would say. I gave you a list of hormones. I gave you a list of the stimuli that causes the enteroendocrine cells to be produced. And I told you what the functions of those hormones were. That is definitely something that, for instance, could be a possible essay question. Would you need to incorporate those into this question? Probably not. Just saying that there are a list of hormones that are released based on the composition would be fine for this description of gastric activity, unless I specifically asked in the question, right, list one hormone that, you know, inhibits the release of activity of the stomach or something along those lines. Maybe if I asked a specific question like that, I don't know how I'd word that or how that would go that, because it's kind of giving away information. But theoretically, if I could think of a clever way to do that, then you might need to do that. But to me, those seem more like two separate types of essay questions. Asking you to identify the hormones that would be released based on a particular stimuli and what the effects of those hormones are, absolutely the possible essay question, right? This, absolutely a possible essay question. But I'm not sure that I would combine the two. That would be tricky to ask, and it involves, it's kind of like the same thing I was talking about earlier. When we started talking about the branches of the enteric plexus and where they're found and what they do, that again sounds more like another essay question to me. So for this exam, for this question, just saying that neural control is reflexes through the enteric plexus seems to make sense to me. Right, that seems to be more appropriate, the same way that you would just say this here. But listing those hormones as another essay question, yes. Listing the branches of the enteric plexus as another essay question, yes, absolutely. Greater omentum is anterior and no. So what I would say is that the lesser omentum is superior. So I think let's take a look at, where's that one picture? So both are related to the stomach. If you remember correctly, the lesser omentum basically connects the liver to the stomach, anchors the liver to the stomach. Ooh, this will work. I like this one. Well, actually, let's start with this one. No, let's start with this one. So notice by picking up the liver, we can see this particular mesentery that anchors the stomach to the liver. And remember, we saw how the lesser omentum connected to the lesser curvature of the stomach. So it is more superior to it. Technically, I guess the greater omentum, because it starts here on the greater curvature and drapes down over the top of the small and the large intestine. I guess it's a little bit more anterior. Notice if we look at this from a sagittal view, right here in dark blue is that representation of the greater omentum going from the greater curvature of the stomach down the abdominal pelvic cavity and then back up to connect to the transverse colon, whereas the lesser omentum is here. So I guess technically it is a little bit posterior and the greater omentum is a little bit more anterior, but I'm not sure that that would be the primary way I would describe them. I think definitely the lesser omentum is more superior above the greater omentum. And then again, it's really their relative positions as well. But yeah, I guess technically the smaller, the lesser is more posterior and the greater is more anterior. Yes, so any organ that is intraperitoneal has a serous membrane on its outer surface. Any organ that is retroperitoneal has an adventitia on its outer surface. That is correct. So if an organ is retroperitoneal, it has an adventitia. If it is intraperitoneal, it has a serosa. So that's why you have one or the other, but you cannot have both.
So adventitia means to be connected to the back wall of the cavity?
I'm not sure if that's what adventitia means, but that is what it does. Notice the duodenum, because it is a retroperitoneal organ, it is anchored in place. It's not going to be able to move and twist around in the abdominal pelvic cavity the same way that the ileum or the jejunum does. The ileum and jejunum can twist and turn because they've got those slippery serous membranes on them, whereas this is anchored in place and not moving. So I don't know if that's necessarily what adventitia means, but that is definitely what the adventitia does. Okay. It anchors it to the posterior wall. All righty. Any other questions? Yes. So when we were talking about the small intestine, yeah, I somewhere in here. So remember when we were talking about the small intestine in lecture, we were talking about how, again, with the presence of proteins, causes gastrin to be produced by the small intestine, stretch of the small intestine causes mucus to be produced, lipids present in the small intestine called cholecystokinin to be produced, glucose called gastric inhibitory peptidase to be produced, too much acidity caused secretin to be produced. And so we talked about all these hormones and all of their functions. And then I also pointed out how your textbook has a chart that does this as well. Talks about a hormone, talks about what it targets, what it does, and talks about the stimulus. So again, cholecystokinin is stimulated by fatty chyme, the presence of fat, but again, some proteins, but mostly it's that fat. And like I said, it tells the liver and the pancreas to produce and release their secretions, releases those enzymes, opens up the hepatopancreatic sphincter, yada, 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 all those things. So like I said, your book had a great table that goes through all of these, and then we talked about them as well. So this is what I was referring to with that for the hormones. Again, the hormones for the small intestine. Yeah, there's other hormones we talked about as well, but this was when we were doing the small intestine, that list for that.
But we didn't talk about histamine.
I briefly mentioned it, but obviously it wasn't one that I emphasized. So my guess is that it's probably not a question I would ask you on the exam.
Okay.
There are more hormones on that list than we talked about in class. So I would focus on the ones we talked about in class. All right. Last call. Any more questions?
Very last one.
Okay.
I'm kind of confused on the intrinsic factor. What exactly is it again? I know it's something to do with B12.
So the intrinsic factor is a protein that is produced by parietal cells and released into as part of the gastric juice. The function of it is basically to trigger the absorption of B12 from your large intestine. Right? The problem is that B12 comes from the breakdown, the bacterial breakdown of certain foods. So it's not present in the stomach. It's not present in the small intestine because those foods haven't been broken down to release the B12 yet. But in our large intestine, the bacteria are able to break down those foods and release the B12, which we need. The problem is, if you remember, the same way we talked about how calcitriol is needed to get calcium to absorb through the wall of the abdominal pelvic cavity, the wall of the alimentary canal, it's the same thing true for B12. You can have a whole bucket worth of B12 in your large intestine, but if you don't have the intrinsic factor, then you can't activate those channels to absorb the intrinsic factor. So with the intrinsic factor present in the large intestine, you can absorb B12, which allows you to make red blood cells. So that's a very vitally important protein, a very vital function of our stomach to make that protein so that eventually we are able to absorb B12. All right.
Simple.
Yep. Sometimes they're simple. All right. Excellent. So as always, if you guys have any more questions, please feel free to email me. I'm happy to answer and respond to those things today or not today, but tomorrow and through the weekend. So study hard. Have a good, safe weekend. We're done in time for you to go get your popcorn and watch the debate tonight. So enjoy that. That should be hopefully entertaining. And be safe, be healthy, be smart. Listen to the doctors. Don't listen to the politicians. Make wise choices. Take care of yourselves and your family, and have a safe weekend. Do good on the exam on Tuesday, and I will see you in class on Thursday. All right. Bye, everyone.